That makes no sense. In this period, humans evolved, doubled their brain size in the length of time that they call a very rapid evolutionary change. Their time span is completely crazy, and they can't study the evolution of anything that's interesting because they study fossils. But in lots of ways, the best rebuttal, the one that most got at these folks advocating punctuated equilibrium, would be the gradualists saying, show me a molecular mechanism. Show me some way in which you can get rapid change and then long stasis, turn that into modern molecular biology, which is occurring 2 minutes after the microevolutionary people were trashing the folks from last week, saying you need to look for the actual genes. It's not enough just to make up stories. And once these folks had assimilated what evolution looks like on the genetic level, mutational level, micro-mutational changes, they loved genetics. They loved the molecular end of it because they could now turn around to the Gouldians and say, show me the genes and show me the mutations that will account for stuff like this, because you can't account for it because we all know classical genetics and mutation, you don't get that, you get gradualism. So this was a period of enormous hostility between the two camps, and the gradualists called the punctuated equilibrium people evolutionary jerks. Haha. And the punctuated equilibrium people called the gradualists creeps. So ultimately, they all got along wonderfully because they were so witty. But what you had was enormously hostile camps, and really quite hostile, because all sorts of implications spreading beyond like how fast the shape of this like seashell was going to be evolving. And when it first came on in the 80s, all of this controversy, the punctuated equilibrium people didn't have a word to say with the show me the molecular mechanisms. Show me mechanisms for mutation that will produce rapid change. And everything that has occurred since then in the world of molecular genetics that has been most striking has supported punctuated models, ways in which the micro-mutational, microevolutionary stuff of an hour ago is not what's going on an awful lot of the time. Okay, starters. So simple classical model. What we've got here is a stretch of DNA, and this box symbolizes a sequence of DNA coding for one gene. And next, right next to it, once it finishes, one of those stop codon, stop triplet signals. Right after that comes the stretch of DNA coding for the next gene and the next gene. And this is what DNA is about. It's the sequence of genes there. And what you would obviously then get is this arrow having this intervening step of that RNA stuff, but eventually producing amino acid that produces a protein of this shape, and this is the protein coded for by this gene, this, and so on, and that's exactly how it works. That's the structure of DNA. Then, though, people began to find that that's not the structure of DNA. What you began to get instead was something vastly more interesting, which is that for starters, when you look at coding for one single gene, it's not necessarily coded for in one continuous stretch of DNA. In other words, it's broken into little pieces, and you will have a stretch of DNA coding for the first third of the protein, and then a bunch of DNA that's got nothing to do with it. Stay tuned. Then coding for the next third, coding for the next third, that the gene was broken up into separate coding domains. And the term that was given for this was, these were called exons, and the in-between boring stuff were called introns. And this was a major finding, which made no sense whatsoever, because how are you going to get from this to then having a protein which has its normal sequence and shape, where this part was coded for by this exon, exon 1 of this gene, and this from exon 2, this from exon 3. That's a completely different sort of world of stuff. How are you going to do that? Because once you're going to make an RNA that's going to encompass all of this, and that's going to code for something completely different, because you got these introns in between there, and people then guessed something had to exist, which was soon discovered, enzymes called splicing enzymes. And what they did was exactly what you need to do to solve this, which is at the RNA level, these splicing enzymes would come along and they would snip out the part corresponding to here, and another would snip out there, and another one as an enzyme catalyzing. You'd stick these two pieces together, and thus you had this utterly bizarre world in which genes, the vast majority of them, are not coded for in a continuous stretch of DNA, but instead are broken up into these separate exons, and then you need these splicing enzymes to clip out the boring in between parts, the introns, stick them all together, and you've got your functional gene. Weird. Okay, but that's how they work, though. And it was shortly after these were discovered that one of the giants in this business, a guy named David Baltimore, who got the Nobel Prize for some of the work on that reverse process of RNA viruses turning back into DNA, he was the first to really appreciate that what you got here is a potential for a lot of information because of, and I think he was the first person to introduce this word into thinking about it, because of the modular construction of genes, because genes come in these separate exons. What does that begin to allow you to do? Very important stuff. Okay, so we have the same structure here, and we have a gene coded for in a modular way in 3 separate exons, and thanks to splicing enzymes, protein catalysts, clipping this out, you wind up with this. What Baltimore was the first, or one of the first to appreciate, was that you could wind up with something different there as well. You could, for example, create a very different protein, one consisting only of A and B, or one consisting only of A and C, or one consisting of B and C, or A alone, and suddenly you have this combinatorial possibility of cranking out a number of different ways of putting together 7 different ways, since you can't transcribe this gene so that there's no transcription, 7 different ways of combining these different exons, 7 different types of proteins you could generate from the same gene. This was not accepted with a whole lot of pleasure by the old guard, because intrinsic in the know the DNA sequence specifies amino acid, protein shape, protein function, intrinsic in that is one gene specifies one protein. One gene only specifies a single protein. One protein is only specified, coded for by one gene, and suddenly this modular business allows you, with one gene, to generate 7 different kinds of proteins. For starters, how could that possibly work that way? Just on a nuts and bolts level, all you need are splicing enzymes that work a little bit differently in different parts of the body. One that will splice this off and is attached to an enzyme that will degrade this, while the splicing enzyme here does, and what have you just produced? You'll get this one, coupling of splicing enzymes with degradative enzymes, and suddenly you've got a means to have tissue-specific expression of genes. The same gene will produce different types of proteins in different parts of the body because of splicing enzymes working differently. Then, just to confuse things even more, people began to note that there would be some splicing enzymes and genes where they would splice at a different rate. And we would now have a gene with A and A prime and other splicing enzymes that would cut at other points, one single sequence of DNA generating all sorts of different types of proteins in different parts of the body, at different times of life, under different circumstances, in different individuals in different ways. Suddenly, there's a lot more information floating around. So that was a huge, huge breakthrough in the field, understanding this modular basis of gene construction. And for our purposes right now, what the most interesting consequence of that is it's no longer one gene, one protein. One gene instead can generate all sorts of different types of proteins, different settings, different circumstances. The next thing that was intrinsic in this model that's now been trashed of one continuous gene, what we just figured out is instead you can have the intron, exons, all of that. The next thing that went down the tubes was looking at, well, how much of DNA is actually devoted to coding for amino acids? And the answer was obvious, like 99.99%. Each one of these would just have to have a stop codon, a stop signal at the end. And otherwise, this was just a continuous flow of information once you have factored in these intron things. Okay, so they're part of this gene, but immediately starts the next one. The next major discovery was one gene would very rarely start immediately after the next one. There would be long stretches of DNA in between that didn't code for a protein, non-coding DNA. That's mighty puzzling. It's like this junk or stuff, and around that time the phrase junk DNA was actually floating around, people trying to make sense of this, and when people sat and started actually like doing the numbers, out came a number that knocked people on their rears. It was so flabbergasting. 95% of DNA is non-coding. 95% does not code for a gene specifying a protein. In other words, in between here, on the average, would be a stretch of DNA 19 times the length of that, or whatever the math winds up being, and suddenly calling that stuff junk DNA started seeming a little bit tenuous, because 95% of your DNA just can't be packing material for the whole thing. It's got to be doing something. And during that period came sort of the insight into this, that all that intervening non-coding stuff was. What was that? That was the instruction booklet. That was the instruction booklet on when to activate these genes. That was the on and off switches for turning genes on or off. Upstream in the non-coding domain, just above a particular gene sequence, would be the information for when that gene is activated into makes RNA, into protein, all of that. And upstream of that are the on and off switches. Implication right there off the bat, which is Crick was wrong. DNA sequences are not the starting point of the central dogma of life, and DNA is the rule giver and all of that. DNA is being regulated, genes are being regulated in some other way, and where 95% of DNA is being devoted to regulation of the genes. DNA has no idea what it's doing. DNA is a readout that's under the control of all sorts of other factors. And out of this emerged the really, really important concepts of regulatory sequences upstream from genes. So here we have a long string of DNA coding for this gene, which happens to come in 2 exons, and it is like the space between a galaxy, how long the non-coding is going to go on until the next gene is back there. So what's going on this stretch just upstream for this gene? Things that were soon being called stuff like promoter sequences or repressor sequences, things, stretches of DNA that coded for switches rather than coding for protein, that coded for things coming in to the neighborhood of the DNA and binding to some of these promoter or repressor sequences, and then turning on, or in some cases off, the transcription of that gene, the process of the gene generating proteins. You would have promoters sitting there, and this is overly literal. It would be just a sequence of DNA, which thanks to that sequence would have a certain subtle micro shape, and along would come something which happened to be able to fit perfectly into that spot. And if and only if this molecule, usually a protein, bound to this promoter, suddenly that would trigger a whole bunch of enzymes to come in, which would start the process of transcribing this gene. This would be the switch, and this is the thing that just turned the switch on. And these things that turn the switches on, or off in some cases, were soon called transcription factors. totally critical concept in there. The fact that vast stretches of DNA don't code for anything. Instead, they have the instruction booklets. Here's how you turn this gene on or off. Send in this molecular messenger, send in this type of transcription factor, and if it shows up, binds to here, you now activate the transcription of this gene, the process of turning that gene, making proteins derived from it. This is where the information was. And this is not DNA knowing what it's doing. This is outside regulators coming in. So immediately, that has made life a whole lot more complicated. Next complications. You could have different genes scattered all over the place that would have the same promoter upstream of it. What would that mean? In comes a transcription factor, and it doesn't activate the transcription of one gene producing one protein. It activates the transcription of a whole bunch of them. In other words, now suddenly we have messengers that could trigger activation of genetic networks, entire arrays of proteins being produced, all with a functional similarity, given by the fact that all of them have the same promoter upstream. So suddenly, you have the possibility of the same promoter being upstream, regulating more than one gene. That is the general rule. Flip side of it, any given gene, for example, could have a bunch of different promoters responding to different types of signals. So now, suddenly, you have this gene which can be transcribed under this circumstance or under this circumstance, and both are ways of turning on activation of that gene. And in this circumstance, the same promoter is found in genes A, B, and C, somewhere at the other end of the chromosome. And in this case, this promoter is found on genes D, E, and F down there, different networks. So the same sort of transcription factor logic, once you can have different promoters upstream of a gene, and once you can have the same promoter upstream from multiple genes, you suddenly have the ability with different transcription factors to activate entire networks of gene expression. Okay, so what this has allowed you to do is completely trash this notion of DNA knows what it's doing. DNA is just a readout, and who knows what's going on? Whatever is controlling the transcription factors, whatever is causing them to do their thing. And this could be a world of influences. And to be absolutely accurate, this requires introducing the word environment. This is environment having something to do with genetic effects. This is going to be a way in which environment is interacting with genetic elements, environment determining which of these are doing what. What would that look like? Sometimes environment could be the environment in the rest of the cell. So you got your DNA there, and your stretches and all of that, and the promoter, and we could do that by now. And something is occurring inside the cell, which activates some transcription factor, and then you go change a genetic event going on in there. The cell is running out of energy. There's various ways to construct sensors. In other words, evolution has come up with a bunch of different ways in which cells can respond to signals of low energy, and that will activate some transcription factor, which will go and bind to a bunch of promoters, which produce proteins involved in taking up more energy from outside the cell, transporting in more, using energy more efficiently. So what we have here is an environmental regulation of genetic effects, environment to the rest of the environment of the cell. The DNA doesn't know what it's doing. The gene doesn't know what it's doing. Events going on elsewhere in the cell is what's regulating what's up. Some of the time, the environment can be even more far-flung. And in this case, it's now events going on outside just this one cell. In this case now, you've got the cell with this DNA. Now, instead, you have a chemical messenger coming from somewhere else. and binds to its receptor like a lock and key. And as a result of this binding, something happens here, which does something here, which does something here, which eventually activates some transcription factor, which goes and does its thing. Now we have gene expression in the cell being regulated by the environment somewhere else in the body. Somewhere else there. What would be a classic example of that? This is what a whole lot of hormones do. Hormones go floating around and they affect cells everywhere throughout the body. By definition, a hormone is a blood-borne chemical messenger. So you could secrete some hormone out of the top of your ear and it will affect things in your little toe, very far flung. Hormones will bind to their receptors, lock and key. Most hormones are protein in nature and trigger what's called a second messenger cascade. Jargon, don't worry about it, just get it conceptually. Activate some transcription factor, deactivate some other and suddenly events going on. 14 counties over there are regulating what proteins are being made in this cell. What would be an example of that? Testosterone, for example. Testosterone secreted from the testes, traveling far and wide and eventually binding to androgen receptors on muscle. And what happens there is, through a pathway like this, turning on the activation of genes, coding for proteins, all sorts of structural proteins that will make that muscle cell bigger. Your muscles are getting bigger thanks to testosterone. Look at this. Events occurring a gazillion cells away in the testes, a messenger here. here determining what gene expression, what gene activation is happening. Sometimes, though, the environment could be completely outside the organism, like that. Sometimes you can have, for example, a messenger from the outside world. What sort of messenger? A scary sight, a bit of sensory information, or whatever. Olfactory. Olfactory, suppose some odorant comes in, a pheromone. You're a female rat who has recently given birth, and the pheromones from your babies come floating in and bind to receptors here. Very similar principle again. And that causes them to activate something, and some cell down there. And eventually, there's a cell here that controls some muscles, and it makes your eyes dilate because you love the smell of your baby, and you're just, I don't know if rats have their eyes dilate, but humans will in many of those circumstances. Aha, how do you do that? You just change some structural proteins that did this or that, or suddenly this female is making some hormone like oxytocin as a result of smelling her baby. What have we got here? We got something going on in the outside world regulating what's going on with genes. Genes as the central dogma of life, as the information giver, nonsense. Events going on in the rest of the cell, the rest of the organism, the rest of the universe are determining when genes activate. So what we see now is a much more interesting world of regulation of gene expression. First, the modular ability for one gene to generate all sorts of different proteins, which brings up an issue after a while is, does that count as one gene? And people argue over that. And because of this 95% regulatory sequence business, the most interesting stuff going on with DNA is not what the protein is like, not what the protein does, not how well the protein does it, but when it does it, in what contexts. And what we've just introduced are if-then clauses into this whole world. If you get a signal that there is low glucose in the cell, then you begin to make proteins related to glucose uptake. If you have the smell of your child come in, then you activate this path. It's not changing what the protein is like, it's changing context. And I think what we will see is context is vastly more interesting than whether this protein is a little bit more like this, a little bit more like that, if it is being expressed instead at a different time, in a different place. And it's stuff here, I don't know what percentage is accounted for by identified promoters and repressors and stuff, but it's a tiny percentage. What that means is there's regulatory stuff going on that no one has a clue about. The main thing, though, here is modulatory structure to genes, introns, exons, and this whole world of the environment regulating when genes are turned on and off, a whole world where you can generate completely different proteins, not a protein that's a little bit more this way, a little bit more that way, in completely different contexts. So all we need to do now is begin to stick this into the molecular biology of mutations and evolutionary change. Where does this begin? Oh, no, before we do that, that's not what we're going to do. We're going to look at one more level of regulation here. DNA, and we already know this whole business, what's telling it what to do, transcription factors coming in, all of that, these interesting implications. DNA, let's see, for our purposes, protected sort of layers of protein that are just sort of structurally stabilizing. This is not really what they look like or quite what they're made of, but they're called chromatin. There's just stuff that stabilizes the DNA because these are wispy little things. And one of the things that, of course, they need to do is they're enwrapped around the DNA stabilizing. You got some transcription factor coming in from somewhere else in the cell. It's got to be able to get down to the DNA. And thus you have a whole world of chromatin opening up to allow transcription factors to get through. And thus you have a whole world of what's telling the chromatin to open up where and when. suddenly a whole world of regulation of whether the transcription factors even have access to the DNA. So you can have tons of a transcription factor and all set to transcribe something off of this, and thanks to conformational changes, folding or unfolding of chromatin, you're regulating whether the transcription factor can even get through. And thus there's a whole world of stuff that changes, chromatin modeling and remodeling. Additional step here, one that's really interesting, is you can do things, circumstances of life, the environment can do things where you change the structure of chromatin around a particular gene in a way that makes it easier to transcribe or harder to transcribe, and you can essentially make that change permanent. You could permanently do something in some particular stretch of chromatin so it will never open up again to allow the transcription factor in. And what you've just done, jargon, is you have silenced that gene. You've silenced it permanently. And people know a lot of the mechanisms for how this occurs. For those who care about such things, the process is called methylation. This is a little bit different. That's occurring with the DNA itself. But this is silencing of genes by structural access of transcription factors to it. When does that occur? There's all sorts of circumstances early in life where you will change the permanent accessibility of some genes and transcription factors. You will cause long-term, lifelong changes. As but one example, and one that we will look at a number of times down the pike there, in rats, the mothering style of the mother rat will cause chromatin changes, permanent ones, in some of the genes related to stress hormones. So that certain types of mothering, how often you lick the baby and other rat mother type stuff, will regulate how readily some genes will be turned on for the rest of your life. This is early experience. This is molecular mechanisms for events early in life lasting forever. A lot of these turn out to be a little bit reversible, but for our purposes, lasting forever, this is a whole new field called epigenetics. Genetics is all about DNA sequences. Epigenetics is all about regulation of access to DNA sequences, things of that sort. So suddenly this epigenetic world is entirely capable of overriding anything going on at the transcription factor end. Just to give you a sense of this, researchers at National Institutes of Health, a guy named Steve Sumi, who studies primate social behavior, and what he has shown is in monkeys, in one part of their brain, you change the style of mothering that that monkey is subject to as a baby, and you will change the conformational access state of 4,000 different genes. Enormously influential there. Enormous arrays of ways of regulating where it's not genetics, it's epigenetics. And this has given rise to a great phrase: fertilization is all about genetics. Development is all about epigenetics. And what epigenetics is about are ways in which the environment not only can regulate what's going on with this gene right now, but can cause lifelong differences in the ability to access genes. So this is an enormous array of levels of regulation, splicing enzymes determining how your exons get mixed and matched, generating all sorts of different proteins, transcription factors representing the things that turn the switches on and off, and the array of switches is far more interesting and plentiful than the gene itself. Transcription factors reflecting what's going on in the outside world, like in the rest of the cell to the other side of the planet, and finally this whole additional level of regulation where some of the regulatory consequences here can be lifelong. Enormous array of levels of regulation and an enormous number of ways in which just the DNA sequence of the gene itself is not very interesting. That determines the shape of the protein. All this other stuff is where is it expressed, when is it expressed, in what contexts, what sort of if-then contingencies, whether it is ever expressed after a certain childhood event. All of that much, more interesting. So what we need to do now is transition to thinking about evolution, mutations, on the level of all the stuff we just heard about. All I will leave you with, and picking up on Wednesday, is think about what if you get a mutation in a splicing enzyme? What if you get a mutation in a transcription factor? What if you get a mutation not in the letters coding for a gene, but coding for a promoter? What happens in all those cases? And suddenly you begin to see a world in which you can get stuff that's not purely gradual.
For more, please visit us at stanford.edu. Stanford University.
Let's see, various announcements. I am out of town tomorrow, so no office hours. I managed effortlessly to confuse a whole bunch of people Monday about positive selection versus stabilizing selection, all of that. On the Q&A part of the courseworks, I've pulled out the paragraphs from the extended readings that explain that coherently, as opposed to what happened here. And Monday, I discovered that somebody sitting in this room, who I cannot spot at the moment, has a spectacular tattoo illustrating the central dogma of life from or at least one person. If there's two, there's really something. So amazing demonstration of DNA to RNA to protein. So if the person doesn't have a class afterward, he will be standing up on top of this counter here, displaying it for everybody's educational purposes. Okay, so besides finding about that guy, what else happened Monday? Monday, we introduced two concepts and with great dramatic foreshadowing started to trash both. The first one being this huge emphasis in all of the preceding lectures on the evolution of behavior, not only this emphasis on adaptation, not only this emphasis on inferring a genetic basis through that roundabout, here's a story and until you make up a better one, I win, but also that emphasis on gradualism, that slow evolutionary change. What we focused on there was what would be the mechanisms, the molecular mechanisms for classic gradualist evolutionary change, microevolution, the whole world of point mutations, deletion, insertion, all of that, the whole world of mutations potentially being big news, knocking a protein completely out of business, and suddenly you got a different gender than you actually are chromosomally, but a world nonetheless where for our sound bite, micromutations are affecting how readily a protein does its job. How strongly, how potently, how long, what its job is, all of that as sort of the grist for the microevolutionary change. What we then transitioned to was the huge attack on the gradualism that came with this view of punctuated equilibrium, this notion that most of the time nothing exciting was happening, long periods of stasis, and then sudden dramatic change proposed by Gould, other evolutionary people. And what we saw was the huge implication there was if most of the time nothing interesting is happening in terms of evolutionary shift, there goes gradualism, there goes the emphasis of every bit of adaptation is going to make a difference long term. Every opportunity to compete and dominate is going to make a difference. All of that goes down the drain if 99% of the time there is instead equilibrium stasis. What we saw were the wildly enthused attacks upon it along the grounds of that stasis and rapid change for paleontologists bears no resemblance to what the world is like for an evolutionary biologist. Paleontologists can only see the evolution of boring stuff, morphology. You miss the entire world of what's going on inside that morphology. And the most damning sort of complaint, show me some molecular mechanisms for these macroevolutionary changes. What we then transitioned to was seeing all the ways in which the picture of the structure of genes and DNA is completely wrong, along the lines of there's the intervening sequences, the intron-exon organization of genes, suddenly the opportunity to mix and match combinatorial abilities for one gene, or at least one stretch of DNA, to specify a whole bunch of different proteins depending on the function of the splicing enzymes, and those splicing enzymes differing in different parts of the body under different circumstances. We then saw that flabbergasting business about 95% of DNA not coding for proteins, not being genes, but instead the instruction manuals, the promoters, the switching on and off, leading to that critical notion of transcription factors transducing events going on out there, outside the nucleus, outside the cell, outside the organism, into changes in DNA, and thus the incredibly important role of transcription factors, promoters, their lock and key interactions, all that sorts of stuff, introducing the second critical concept. If microevolution is about sort of changing the function of a protein, changes occurring at the level of transcription factors, the promoters, those are about changing the context for which proteins are functioning when genes are expressing. What they are about are introducing if-then clauses. If this is happening outside the nucleus, cell, universe, whatever, then if that results in this gene being activated, then you will see that response. What came along sort of de facto with that, the second major thing that we thus trashed was DNA as the central starting point, the dogma of life. DNA is the only one who knows what's going on out there. DNA commands, RNA commands, protein commands all of life. And what we saw was DNA genes are just a readout, and most of what it's about is environmental regulation of when genes activate. Finally, we began to see all sorts of ways of futzing around with gene expression that has nothing to do with the sequences of DNA, but instead has to do with things like access of transcription factors to the DNA, changing accessibility permanently, that whole world of epistatic, epigenetic changes, coming up with that soundbite. Fertilization is about genetics. Development is about epigenetics, all of that combining to show, number one, genes are not such a hot deal in terms of them knowing what's up. Number two, in addition to mechanisms known for microevolutionary change, we left it just on the cusp of seeing how all of this stuff can set you up for some big-time macroevolutionary changes. First example of it, first way in which that could occur. So back to the modular construction of genes, we've got our introns, exons, all of that. And what we learned the other day is here we've got a gene coded for with 3 exons. You produce a messenger RNA that encompasses all of these plus the introns. Along comes a specific splicing enzyme, knocks out these parts, and here is the mature protein. So that's great. What if you have a mutation in your splicing factor, in your splicing enzyme, and as a result, instead of clipping here, it ignores those and clips here instead? Or what do you get? You get 2 completely novel proteins that never existed before in this cell, in this individual. What you've got there is not some sort of little microevolutionary change. This is not a protein working a little bit better or a little bit more sluggish. This is the invention of entirely new proteins. So what we see here is splicing factors working differently, and this is a major change if whatever went on in the outside world that causes the then of this being made, if you've got that mutation in the splicing factor, you've just made an entirely new if-then. If whatever's going on in the outside world happens to activate this, you produce these 2 novel proteins. So we've suddenly got the potential for producing all sorts of novelty there. Splicing enzymes, proteins, thus there are genes coding for the splicing enzymes, and as a result, suddenly a very different type of consequence. Yeah, question? Great question. How often do they make their way out of the, what was that? Do they have any function? Okay, that's a critical question. Wait about an hour and 17 minutes and you will get the answer to it, the answer being not very often. Oops, I gave it away. The answer being not very often, and we're going to see why that's real critical to punctuated equilibrium. Okay, so we see one realm of major consequence, and intrinsic in that is, wait a second, okay, so if splicing enzymes are genes, there's a gene for the splicing enzyme, and that gene has its promoters, and that gene maybe is in a number of exons, and thus it needs a splicing factor. And we're rolling all the way down their recursive regulation. So one splicing factor, big consequence. The next realm where you can have a big consequence is now you got a mutation in a promoter. A whole world of a mutation in a non-coding part of your DNA, and you can immediately run with that one. Have a different promoter, and it's going to interact with a different transcription factor. And we go back to that business about typically a promoter, the same version of it, multiple copies of that promoter appear upstream of various different genes, promoters mediating expression of entire networks of proteins. Change that promoter, and you're going to change the network. Change that promoter in only some of the places where it occurs, and now you've created a completely novel network, a network consisting maybe of half of the proteins that you would have made in the unmutated version. Mutate every single version of it, and maybe it's an entirely different transcription factor that interacts with that promoter. And thus we've made an entirely new if-then clause. Not just a new if-then make never seen before protein. Now it's an if this happens, then make a network of proteins that have never existed before, a combination of these that have never occurred before. What we're beginning to see here is a theme of amplifying effects. Huge major consequences. Instead of one little protein, which thanks to one little base pair changing is 1.5 degrees more folded this way instead of that way, and 1.5% better at binding this or that hormone. That's the microevolutionary change. This is setting up for big network changes, novel genes, novel networks, novel if-then clauses. Third range, okay, an example of why promoters and promoter mutations are interesting. Later on the course, we are going to hear about one of the all-time interesting differences that you can have in one of your promoters if you happen to be a vole, and increasingly it's turning out equally interesting if you happen to be a human. This is a promoter upstream of a gene having to do with the hormone vasopressin. Do not panic or care if you haven't heard of this yet, but you will have in more detail within a few weeks. Vasopressin is this hormone which has something or other to do with social affiliative behavior in males and all sorts of interesting stuff with that. And naturally, it being a hormone out there is a vasopressin receptor, and thus there's a vasopressin receptor gene, and there's a promoter upstream of that gene, which turns out to come in a couple of different flavors. And you look at voles, which are little hamster thingy sort of things, and there's all sorts of different vole species, and there's ones in the mountains, and there's ones in the plains, and there's ones in the, from California to the New York Islands, and there's all these different species. And it happens, some of them happen to be monogamous, some of them happen to be polygamous. We're often running with the sociobiology of that and how many imprinted genes and all that they're going to have. But a critical difference in monogamous vole species, there is a different promoter upstream of the vasopressin receptor gene than you find in the polygamous ones. And go and mess around with them, use gene therapy techniques to change the promoter, to modify it, and you could convert a polygamous male vole into a resoundingly monogamous one. And I don't know if this counts as gene therapy, like curing a disease, or just gene transfer sort of stuff, but what you've got here is change your promoter, and you suddenly have a different pattern of expression, which parts of the brain it winds up in. Suddenly you have made a major shift in behavior. You're not causing a change in a gene, you're causing a change in its promoter. You've just changed a major if-then clause. Another example of it, there's a gene that codes for a hormone neurotransmitter that has something to do with pain perception called dynorphin. It's broadly related to things like morphine and such, and it's got a dynorphin gene that's a little more complicated than that, but there's a promoter upstream. And recent research is showing that the number of copies of that promoter in different rats predicts something about how readily they become addicted to various drugs. And that winds up being pertinent. Oh, back for a second, back to the vasopressin gene and the vasopressin reporter gene and its promoter. In the last couple of years, a number of studies come out. One, for example, in a very credible journal by a great group showing that if you happen to be a human male, which version of that promoter you have gives you a certain significant predictive power over how stable your social relationships are going to be. Get a load of that one. And that one is coming later on. Whoa, have a different type of promoter, and statistically, you are more likely to get divorced down the line. Okay, back to that first lecture, free will stuff. There is so much more to come along those lines. We will look at the vasopressin system much more in the sex lectures, but again, that's not a difference in a gene, that's a difference in a promoter. Next domain, we've now seen changes, subtle changes, dramatic changes in splicing enzymes and the genes underlying them, changes in promoters. Next obvious domain where you can get a macro change is a mutation in a gene for a transcription factor, obviously. Do that and you're going to have completely different networks. Once again, you will have changed if-then contingencies dramatically. Transcription factors are very, very important, obviously. One measure of this, when you look at the human genome versus the chimp genome, and we will eventually do that in some detail, and say, okay, 98% of our genes in common, which is kind of accurate, and you remember from the other day how that's different at level of explanation than you sharing 50% of your genes with your full sibling, all of that, back to us sharing 90%, 98% of our DNA with chimps. What are the differences? And there's going to be some really interesting differences we're going to focus on down the line, but one of the patterns that's come out of that is a disproportionate share of the genetic differences between humans and chimps are genes that code for transcription factors. And that makes perfect sense. You get a change in some gene coding for some structural protein, and maybe your muscles will like bend a little bit this way instead of that way, or who knows what. You get a change in the gene coding for a transcription factor, and you will have invented all sorts of novel networks. So a disproportionate share of what has gone on in evolution differentiating us from chimps are changes in transcription factors. This was a big triumph, big support for a view that came out in the 80s. There were parascientists at Berkeley, King and Wilson, and this iconic set of studies they did. They were the first people to come up with the 98% business there, and using very primitive molecular techniques that have since been affirmed. And they also came up with a prediction, a purely theoretical one, which is the most interesting changes that will occur in evolution are in regulatory parts of DNA rather than in the coding parts for protein. And everything since then has supported that, including things like you want to turn an ancestor of a human and a chimp into something that will look a whole lot different from a chimp, turn into us, change stuff with regulation, transcription factors, promoters, etc. What you see with that is this is endlessly networks for amplifying effects for macro changes. And there's a new person in the department, bio department, Hunter Fraser, a new assistant professor who works in this area, showing just how much evolutionary change is being driven by changes in regulatory parts of the DNA world rather than the genes itself, changing a little bit about how this protein works. That's okay. Changing if-then clauses for entire networks, that's hugely important. Another interesting factoid, there's now been the genomes of, I don't know, about 100 different species sequenced, and ranging from really short genomes with, I don't know how many how many genes in there to the longest ones. When you pile them up and you look at them as a function of how long their genomes are, the more genes you find in a species, the greater the percentage those genes are that are transcription factors. And this makes wonderful sense as well. You got one gene, and you only need one transcription factor. You got two genes, and you could milk maximal information out of them with 3 transcription factors. You transcribe A, or B, or AB. You've got 3 genes, and there's 7, and the equation is 2 to the nth of this one, or whatever. But what you see is, by 4 genes, you're up to 15. By 5 genes, you're up to 30. You're having an exponential, a dramatic increase in the number of transcription factors you need to take advantage of all the possible combinations of networks of gene expression. The larger the number of genes you find in an organism, the greater the percentage it is of transcription factors. In other words, get tiny little micro changes in DNA coding for transcription factors, splicing enzymes, promoters, and you're going to have big major consequences. So over and over here, we see this contrast. Microevolution is about the function of proteins. Macroevolution is about which proteins, when, networks, if-then clauses, far more consequential. Okay, one additional domain of, we're not talking about a tiny little micro change in one base pair, one additional domain, a highly revolutionary one that came some years ago, that also has lots of implications for thinking about macro stuff in evolution. And this revolved around one of the great irresistible, musical dramas that have ever come out of the history of science. Ones that is, there's got to be a musical somewhere in the future of this, having to do with a scientist named Barbara McClintock. Barbara McClintock, if you are sort of a modern molecular sort of person, at some point or other you will have to have sacrificed a goat at the altar of Barbara McClintock. She is so amazing, and she has such a stirring story as to what happened with her, one which actually is so. Okay, Barbara McClintock was born, I don't know, about 1900 or so, and was a plant geneticist, and was sort of off doing genetics of plants and doing something or other with maize, whatever those people do. And she was extremely successful. She was wildly successful, I think at age 40 or so in the late 1930s. She was already a member of what's called the National Academy of Sciences, which is the most honorific science club you can belong to in this country. 40-year-old women in 1940 were not becoming members of the National Academy of Sciences. 110-year-old white guys with shiny foreheads were becoming members of the National Academy. For her to have been elected, at that point, she was an amazing scientist, one of the absolute leaders in the field. So she's cruising along there, being renowned, and one day she made a discovery that completely destroyed her career. So she's sitting there one day, and she studies beloved corn, maize, and patterns of inheritance based on colors of kernels, genes, molecular biology, non-existent, none of the stuff available now. All you could do in terms of making sense of what is being inherited, patterns of inheritance, is just looking at the phenotype, looking at the appearance, peas and whether they're wrinkly or not, people and whether they're wrinkly or not, corn and what the colors are of the various kernels there. That was cutting edge molecular sort of genetics at the time. Okay, so she's working in that domain, and she's seeing a result. She's seeing a pattern of inheritance which keeps popping up in certain circumstances, and you go through all of the inferential math that was available, and you crunch through everything, and you, if you were Barbara McClintock pursuing this, you come up with a conclusion that's totally nutty. The only way to explain how this change was occurring was if genes were picking up and moving around on the DNA. If genes were jumping around, if genes were mobile, moving around. And out of this, she came up with a proposal that there are such things as transposable genes, transposable genetic elements, transposons. All of the people who spend decades afterward mocking her in various ridiculing tones of voices soon refer to these as jumping genes. And the general consensus in the field was that Barbara McClintock had gone out of her mind. Yeah, genes jumping around. Yeah, right. I'll sell you the Brooklyn Bridge after that. That's ridiculous. ridiculous. That's ridiculous. And Barbara McClintock, having a certain stoic, self-esteem, sort of based personality of one that was basically said, you know what, this is what I see. You want to believe it, believe it. You don't want to believe it, don't believe it. Leave me alone. I want to go back to my experiments. And she essentially disappeared from the field and just sat out in her cornfield at a lab in Long Island called Cold Spring Harbor Lab and just chugged along on her own for decades afterward. She wrote papers about it that were incomprehensible to people because no one could believe anything this ridiculous. And she was developing this whole story about genes that move, transposable genetic elements. Everybody ignored her. She was mocked. She was pilloried. She was burned at the stake, all of that. And then finally, somewhere in the 1980s, molecular techniques caught up enough to show she was absolutely right. And these things now are called transposons. Transposable genetic elements, genes really do pick up and move around. And this was an amazing landmark discovery. The entire world went crazy about Barbara McClintock at the time. She was on the cover of like post-meeting boxes of cereal. There were Barbara McClintock like lines of like, you know, dance clothes and exercise videos and recipe books and all of that. And Barbara, and oh, somewhere along the way they gave her her Nobel Prize, and she was in her late 80s at the time, and showing exactly the stuff she was made out of. She said, well, that's nice, thanks for the Nobel Prize, but you know what? I didn't really need to have gotten it. This is what I saw. You want to believe it, yes or no? You believe it now? That's nice. Leave me alone. Let me go back to work. And she continued to work in her cornfields doing experiments up until about a week before her death in the early 90s. This is a totally cool, amazing figure in the history of science, a lonely pioneer. As it turned out, she wasn't that lonely of a pioneer, and people didn't think she was quite that crazy. And apparently a lot of her papers were ignored because she could not write and her papers were incomprehensible. But nonetheless, the general picture was she discovered all of this on her own, staked her career on this, and most people thought she was out of her mind and eventually vindicated. Totally cool sort of piece of the history of science. Really, really inspirational person. And I met her once and got to like see her with her corn, and she was like she was like 90 at the time and a remarkably like nice, low-key person, where after about 13 and a half seconds it was obvious that what she mostly wanted was for me to get the hell out of there so she can go back to her corn, which was her response to everybody there, but very heroic figure. So she discovers this entire new world of these transposable genetic elements. And people have been studying it since, these jumping genes. The first thing that has become clear as the field has matured is she picked the right species to study. She never would have discovered transposable genetic elements if she was out there alone in her cornfield studying, you know, sperm whales or something. You know, separate of the funding problems and the logistics, she would not have found it. She did it in the right organisms, which were plants. Okay, think about it. You are an animal, and one of the things you can do is when the going gets tough, you can get up and run away, or you can crawl away or fly away, or whatever sort of animate animal type things can do. If you're a plant, you're stuck there. You can't run away. And if you're going to survive a challenge, you're going to have something more subtle going for you than, ooh, let's run and get out of here. And it turns out all sorts of realms of plant stress responses are in just the avant-garde of molecular biology. Plants have to have fancier tricks than all sorts of boring animals because plants don't run away. What they do instead, among their various defenses, when a challenge, a pathogen, a climate change, whatever it is, comes along, one of the things they do is there's realms of their DNA where they move genes around, where they shuffle stuff around in the hopes of stumbling onto something novel and useful to get them out of that mess. Plants have inducible events of genes moving transposable genetic elements. They tend to induce them when the plant is under some sort of challenge, a cellular stress response. And the way that's done is by activating an enzyme called transposase. And those of you who are new to the business, enzymes tend to have ASE at the end of the word, lactase, sucrase, transposase. And what you've got there is, this is a defense on the part of the plant. Juggle some of its DNA prudently and see if you could come up with something to help you. Make a copy of the gene and then go plunk it down somewhere else and see if you've stumbled into something useful. And it was only in the aftermath that people started to look at the same issue in animals and vertebrates and mammals. And shockingly to everyone, except sort of people who hung out with her, was the fact that we've got them too. We've got transposable genetic elements, we animals. We've got them where they were first discovered, made a lot of sense as well. Okay, you are some scientist and you've just invented in your lab some pathogen, some toxin, some who knows what that has never been seen before in the history of the planet. You've synthesized it and you inject it in a whole bunch of people and they get totally sick and miserable. And then you come back 2 weeks later or 2 months later and they will have made antibodies against that thing. Their body, their immune systems will have made antibodies against some invasive pathogen thingy that never existed before in the history of the planet. And a staggering challenge is, how does the immune system come up with this vast variability for dealing with novel pathogens, making antibodies that will recognize them? And people soon discovered one of the tricks was splicing up genes relevant to making antibodies and juggling them around, inducible transposable events, in the hope of making a gazillion new types of antibodies in a remarkable filtering process that goes on in the immune system, spotting are any of them good against this new thing that just showed up. That's where you had a lot of transposable events in the vertebrate immune system in response to novel pathogens. Turns out we weren't the only ones doing that, because there were other things that could be happening. There were all sorts of the pathogens that could do the same exact thing. There's one tropical parasite, trypanosome, which is one you do not want to get. And trypanosomiasis is the inflammatory disease you get from a trypanosome, this parasite, and it shows up in your body, and your body does this inducible trick, and thank God comes up with some antibodies that could begin to target it and attack it. But trypanosomes also worship at the altar of Barbara McClintock. What they do is, a couple of weeks into it, they take away the surface proteins on their surface, and they juggle some of the relevant DNA and come up with a novel version of it. So just as you've got the antibodies online, you can't recognize the thing with those antibodies. You've got to start the process all over again. And thus, trypanosomes are always a couple of steps ahead. Thus, the immune system has to have evolved better ways of juggling, coming up with novel stuff, co-evolutionary races there. But the cornerstone of it is inducing movable genetic elements. And what people have learned since then is it occurs outside of just the immune system and under interesting circumstances. One really amazing one, which I was going to tell you later, but I will tell you now because I just can't wait. There's one transposable element that's very predominant in primates, and there's a certain cell type and a certain time of life when it is most mobile, when it moves around the most, which is the cells in your brain that are going to be making you new neurons, neural progenitor cells, at the time that they start proliferating and making new neurons. There is an inducible event at that time where you increase the movement of that one genetic element. What are you doing? You're making some new neurons, and as it turns out, in the fairly controlled realm of your DNA, you decide to shuffle the deck a little bit, just because you want to get interesting, novel sort of things that neurons can do. This is totally amazing. This is totally amazing because, among other things, what this tells you is the cells in your body that have the greatest thing to do with making you who you are the least constrained by genetic determinism, because right when these types of cells, neurons, are first being generated, they're doing more shuffling of genetic cards than any other cell type in the body. That sure takes away the power of genes a lot when it comes to the nervous system. These transposable events make a whole lot of variability, some of which is wonderful. some of which is not. And thus, coming back to the question that was asked before, are these disastrous in most of the cases? Yes, but in a few minutes we'll see exactly why. It is not likely that when you randomly shuffle a bunch of cards, they're going to come out in a perfect, sequence of numbers or some such thing. It is a long shot to get something interesting out of it. Nonetheless, this is a mechanism for doing this. What this allows you to do by moving parts of DNA around, making a copy of this stretch, and then moving it, and I don't know the field that well, but I think at least in most cases, the notion is it plunks down randomly somewhere else in the genome. By moving stuff around, you can have big macro consequences. For example, suppose you've got, by now, an if-then clause, introducing this concept already, if-then clause, and we can translate this totally primitively into worlds of promoters and worlds of the actual gene. Okay, so you've got an if-then clause. Suppose you are dehydrated. If you were dehydrated, translating that into actual biology, I don't know, your hematocrit or how wrinkly your kidneys are getting or some such thing, then tell your kidneys to start doing something or other that kidneys do to retain water, which I once understood for a final but no longer do. But you wake them up and they have some response. So we've got an if-then clause. If you are getting dehydrated, then make your kidneys work in a way that increases water retention. This is ridiculous. There is no, if you're getting dehydrated promoter, there is no gene that is equivalent. There's networks though. There's networks and there's ways in which your kidney monitors that and other outposts in your body. So we have a rough if-then clause. Wouldn't take a whole lot of imagination to turn that into real biology. Now suppose you have some transposable event. Suppose whatever it is, the promoter world of that picks up and moves, and the if you were dehydrated part of the if-then clause floats around. It gets plunked down upstream from the genes that say, go and ovulate. What have you just invented? What does this allow you to do? So now you've got an if-then clause, a promoter that responds to dehydration, and it turns on genes related to ovulation. What does that get for you? Any ideas? Oh, come on. Ovulate really frequently. Okay, depends on your threshold. If like you should skip orange juice in the morning, does that mean you're dehydrated enough to ovulate? Okay, you could set it at a very low threshold like that. You could do that. You could do something else though. What else? Yeah. Okay, you're about to die of dehydration, and that gives you one last chance for a round of passing on copies of your genes. If you could find some guy who isn't dehydrated to a dramatic blood flow extent. Okay, that's a possibility. What else can you do? Yes, seasonal mating. That's what it's mostly used for. Certainly possibilities here, but what this allows you to, you're a species where 6 months of the year it's dry and 6 months of the year it's wonderful, wet and lush and exactly the time you want to be having a baby, and you've got like a 6-month gestation period. What do you want? You want your body to know when it's the dry season, and that's the signal to mate because you want to give birth during the rainy season. And then there's species where, you are pregnant for 2 weeks or so, and what you want to have there as a rule is, oh my God, if it's the dry season, don't ovulate, because I'm going to give birth to kids who are going to starve or some such thing. Let's wait until I get a signal that I'm totally wet and hydrated, then ovulate. We introduce novel if-then clauses, and for certain species, this would be how you would do seasonal mating, how to know you should ovulate at a time of year where, given your gestation length, it's going to set you up for giving birth at the time of year when your offspring are most likely to survive, and thus you'll have passed on copies of your genes, all of that. So that's great. Another example, and this one's immediately accessible and is a first crude way of beginning to approach some of the stuff that just flowed seamlessly and cheaply in the forms of theories from last week's stuff. Okay, so you have some if-then element in there and a promoter, which in some way can tell this individual near me smells like me. And we will see by next week exactly how that translates into genetics. But this individual kind of smells a lot like me, and it has an if-then clause which immediately shuts down transcription of all sorts of things related to fertility and so you don't mate with relatives. Some sort of incest taboo runs through a gazillion species out there. Individuals smelling like you, if you are a hamster, make you much less likely to mate with them. So very logical if-then clause. All of that works great. And now you've had a transposable genetic event, and you plunk down the if it smells like me, then into upstream of the gene that says cooperate. And what have we just invented? The starts of kin selection, and you could see all sorts of rules like if you have more promoters, you could begin to have subtleties of saying if they really, really smell a lot like me, like if all these promoters are going off at once, then really, really, really cooperate. If they smell only somewhat like me, only somewhat cooperate, you could begin to fine tune that. What have you just invented? A way of taking sensory information about degree of relatedness and turning that into your extent of sacrificing for one sibling or 8 cousins. So you could begin to see how you invent new if-then clauses. Obviously, this is ridiculous. Obviously, this bears no relationship to what's going on in the real world. This would be happening in nose cells. This is occurring down in the ovaries. There is no promoter that responds to, ooh, somebody here is smelling like me. But there's some that do stuff not all that far from that. You could begin to imagine turning this into real biology, how you could program for this. And once you've got genes moving around, you've plunked a promoter down someplace else, you've just made a new if-then clause. Now, the possibilities of transposable genetic elements also raises the possibility of moving around parts of genes, not just parts of regulatory elements, moving around parts of genes. How would you do that? How would you move a part of a gene? Exons, that's that modular construction of genes again, where if you get a transposase that comes and does its thing there, and in making a copy of this, moves this stretch around, you're now moving copies of parts of genes around, and you can create new genes. For example, here we have the basic mechanism of action of steroid hormones. You guys who need an introduction to that, we'll get it in a week or two. Steroid hormones, hormones like estrogen, progesterone, testosterone, glucocorticoids, all of these, they work as follows. Steroid hormones can enter a target cell and they bind to their receptor. Yes, indeed, lock and key, all of that. It happens, steroid hormones are not made of amino acids. They have a different structure, but nonetheless, each has a distinctive shape, and each type of receptor for a type of steroid hormone has a distinctive shape driven by its amino acid sequence and thus gene code, all of that. So you've got a specific type of steroid hormone fitting into its specific type of receptor, estrogen into an estrogen receptor, that sort of thing. And what it does as a result is it activates this receptor complex. And on the other side of it is a conformation which recognizes a particular promoter down on the DNA. In this case, what would be called an estrogen responsive promoter. So what have you got there? You've got events going on in the outside world. You are like reading the right parts of some novel, and suddenly you're secreting certain hormones that weren't there before, and you're changing genomic effects shortly afterward. This is environment regulating genes like crazy. What is this requiring? One part of the receptor recognizing the hormone specifically, and one part of the receptor recognizing its specific appropriate promoter. So now along comes one of those transposable events, and it happens steroid hormones. This would be called the hormone binding domain, and this would be called the DNA binding domain. In steroid receptor genes, those are in different exons, and suppose along comes a transposable event, and you clip this part off and you stick in a different hormone binding domain. So you've just made a completely different if-then clause. If this hormone is around, then do this. Now suddenly instead it's, if this hormone is around, then do this. New if-then clause. Here would be one possibility. Okay, one of the class of steroid hormones, glucocorticoids, which eventually you will come to love because you will hear endlessly about it in that zebra book. Glucocorticoids, they're stress hormones, human version hydrocortisone. For our purposes right now, what's interesting about them is they suppress the immune system. These are steroidal anti-inflammatories. When you're taking non-steroidals, you're taking things that work like glucocorticoids on the immune system, but they don't have some of the side effects. Glucocorticoids suppress the immune system. It is very well understood, and glucocorticoids come in and bind to the hormone binding domain of the glucocorticoid receptor, and this translocates to a glucocorticoid responsive promoter. That's its whole thing. So now you have gotten a transposable event, and what you've done instead is plunked down the hormone binding domain from the progesterone receptor. So suddenly you got an if-then clause that's novel. Instead of if there are glucocorticoids around, suppress immunity, now instead you've got if there's progesterone around, suppress immunity. What do you think you've just invented? Any uses for that? Sort of, if you happen to know sort of what progesterone is about and where that might have been a great invention to come up with. Any speculations? During pregnancy, okay, progesterone, which is progestational. Okay, so you suppress your immune system during pregnancy. How come? Why is that a clever thing to do? Yeah, instead of... Did you just say so your body doesn't eat your baby? Okay, well, there you go. I don't know what your family is like, but I won't speculate. But yeah, you do that so your body won't eat your baby. So your body doesn't do that so that you don't have an immune reaction against it, and that's a whole world of having to decide this thing belongs here instead of this thing having invaded my placenta. Back to that word that was used by gynecologists talking about last week, the imprinted genes, male-derived imprinted genes making for more potent invasion into the placenta, all of that. Yeah, this is a great way to now do that deal of you suppress immunity during pregnancy, you are less likely to have some weird immune attack on your fetus. And that was a great invention. That was a wonderful thing to have come up with. There's an interesting consequence of that, though, one that pops up in medicine often, which is, okay, so you are immune suppressed because you're pregnant, and then you give birth, and you stop being immune suppressed. The progesterone disappears for the most part. You're off into a very different endocrine world at that point. Your immune system comes back to where it was beforehand. But there's a potential problem, which is your immune system is so wiped out and you're so distracted changing diapers and like having no sleep whatsoever, and sort of your immune system is a little bit out of control. What if it recovers from this pregnancy immune suppression, and instead of coming back to where it was before, it overshoots a little bit? What have you gotten at that point? Okay, your immune system shoots into being more active than it should be at that point. What class of diseases are you set up for now? Autoimmune disease. Whoa, that's as good as the lock and key, everybody knowing that one. You are set up for an autoimmune disease then. There's a whole realm of autoimmune diseases that tend to have either initial onset or flare up in the post-parturition period for women. And in fact, there's some autoimmune diseases, a very serious form, say of lupus. Women really should not get pregnant because they are going to get such a burst of lupus sort of flare-ups afterward. So this is a clever thing during pregnancy. As usual, you don't want to overdo it. Our main point here, though, is by doing some transposable event, moving one exon around and sticking it to another exon that never existed before, you've made up a completely novel if-then clause. Okay, so where have we gotten so far? We are worlds away from the dull, gradualist world of micro-mutational stuff from the other day of one protein working differently. You have the capacity to invent completely novel proteins through splicing enzyme changes, through transposable events. You have the opportunity to make completely novel networks with mutations in promoters, with mutations in transcription factors. What all of this means is there's going to be major changes. And thus, after a 5-minute break, we will come back to your question there of what are the odds of these changes actually being good for you? Okay, so 5-minute break. scientists, sometimes you will find a gene will be duplicated. There will be an extra copy of the gene. There will be 2 copies, one after the other. Gene duplication. Sometimes you can have even a larger expansion of the number of copies of a gene, or you can have duplication of a whole stretch of genes. And this is falling into this new area that people refer to calling it copy number variant, and ranging from one extra copy of one gene to massive duplication of whole stretches of chromosomes. And no surprise, you wind up getting interestingly different things going on at that point. What we'll see later on in the course is there's more and more evidence that the disease schizophrenia involves mutations in copy number variants. And here, this is not a mutation in one base pair. This is not a mutation in one transcription. This is like extra copies of genes sitting there. This can have some very interesting implications. In some cases, the second gene can function as a backup. If something goes wrong in the first one, there's a second one there doing its job, and there's some suggestion that something like that is occurring in some subsets of Alzheimer's disease. Or what you can have is the number of copies of the gene you make has something to do with how much of the protein you make. And there's recent studies showing that when you compare Japanese populations with Western European ones, on the average, Japanese populations have more copies of a gene that has multiple copies, more copies of a gene that makes an enzyme related to carbohydrate digestion. I have no idea what the implications would be of that, but this is not a populational difference in a DNA sequence or in a protein. This is simply the number of copies of a gene. What a second copy, what a duplication also allows you to do in the most metaphorical sense is experiment with one of the copies, because the other one is there taking care of whatever the function is that's critical. What you will see is you get faster evolution going on with genes that you have duplicated, where one of them is the one that in a sense is freed to have more dramatic movement, and what you then see is it's more likely to stumble into some great use without sacrificing the initial use in the process. And there's a guy at the University of Oregon named Joe Thornton who's done really interesting work on the evolution of genes for steroid receptors. And what he has shown from ancestral genes is that's exactly what's occurred. A lot of what are now 2 different genes for 2 different types of steroid receptors were once duplicates of the same gene, and one was allowed to float and eventually, in at least some cases, stumbled into something useful while the other one held into place. In passing, what that phenomenon does is help explain one of the endless, frustrating, exasperating, irritating things that people who attack the notion of evolution bring up, which is the famed soundbite that they have of the problem of irreducible complexity. It always runs the same way, which is saying evolution can't possibly exist because what good is half of an eye? You've got to have those intermediate forms, and what good is it? You couldn't have invented an eye. Evolution could not have produced an eye in one mutation, one generation, and thus it would have to be in a series of steps. And what good are the series of steps? They can't exist. There can't be anything such as evolution. Off you go, hallelujah. So what you get here in these cases is a demonstration instead by having extra copies of genes, one of which is freed to be evolving. You don't have to have a rapid transition from one to the other. You can have this thing moving along, stumbling along, until it just happens to come up with a shape of a receptor that just happens to be able to bind a hormone that stumbled its own way into existence 10,000 generations earlier, which, because it was duplicated, it didn't matter that one copy was now of a form where there was no receptor on Earth for it until it happened to stumble into that. And there's more and more evidence that duplicated genes have a way of describing these intermediate states where you don't necessarily have half an eye, but instead you have the pieces ready in place there for when one thing suddenly pops up which completes the picture. In fact, you can have, as it turns out, sort of half an eye. Russ Fernald in the biology department has done really cool research on the evolution of eyes, and you should read about it sometime to read basically how eyes evolved from like a single layer of cells on the surface of some ancient proto-something, and you sure can have half an eye and a gazillion intermediate forms. Nonetheless, this business of multiple copies allowing you the freedom to have looser evolving of single genes at a time, critical mechanism. Okay, so now we've got all of our pieces in place. Changes in splicing enzymes and promoters and transcription factors and transposable elements and number of copies of genes, number of copies of whole stretches of genes. What wind up being the consequences of this? Back to the question brought up before. So what we saw the other day is sometime futz around with one single base pair of one single gene, and you've got somebody who's going to be dead at 3 months of age. PKU, phenylketonuria, one single gene could be a total, utter disaster. My God, instead now, thanks to these macroevolutionary changes, you change one single base pair, and as a result, you change 7 different genes. No, not 7, but you change more than one gene. You have one single base pair change, and if it's an exon that's used in a lot of different combinatorial ways, you've now produced mutated versions of a whole bunch of different proteins. One single change in a transcription factor, and you've invented an entirely new network of expression. What are the odds of stumbling into something there that is going to suddenly be great and wonderful in all those different areas of consequence? It's really unlikely. What have we just seen here? This is a very stabilizing mechanism for equilibrium. Equilibrium, long periods of stasis. What we've got is if a single base pair change is going to affect half a dozen different proteins or affect through a transcription factor entire networks, the odds are pretty lousy that you are just going to happen to stumble into one that works in all those domains, or at least works in enough of them that it doesn't do you in others. Most of these macro mutational changes are going to be bad news. Most of the time, in other words, there is stabilizing selection against macromutational changes. So what have we just gotten? We've just gotten a straight line. We've just gotten long periods of stasis. So when do the changes come? When you have some circumstance that is extreme enough that it doesn't matter that if this mutation, you've made 11D new types of proteins and 11D minus one of them are not great news, and the final one used to kill you. If the final one now is the trait that is going to save you, it is going to carry the weight of all the other proteins that are changed. If you get what is called an evolutionary bottleneck, if you get a circumstance of such severe selection for such a tiny subset of traits that basically the rule is it doesn't matter. If you have that trait, it doesn't matter how much of a network you've changed, you're going to be one of the ones who come through and everybody else does not. And the evolutionary record is full of all sorts of circumstances where there have been selective bottlenecks where like 1% of a population comes out the other end of it because of some rare trait that they had, which carries all the macro consequences of that. For example, there was clearly some sort of bottleneck of selection that went on with cheetahs about, I don't know, a couple of thousand years ago or so. People have estimated more accurately, because all cheetahs are so genetically similar to each other that you can transplant tissue, you can graft tissue from any cheetah on Earth onto another one, and there will not be rejection. All of the cheetah on Earth are closely related descendants of what had to have been fairly recently, a couple of thousand years, a tidy remnant population that now is highly inbred, and you had some sort of selective bottleneck that went on at that time. Similarly, I can't remember when in hominid history, but the suggestion was at least a couple of points in hominid evolution, there have been points of selective bottlenecks where glaciers or comets or who knows what, where only a small subset of individuals with some traits that have been up until then neutral or unlikely to be useful because of these big macro consequences, suddenly only the folks who had it came out the other end. What are we beginning to describe here? Circumstances of rapid change. Circumstances of punctuated radical change there. Circumstances that begin to make conceivable punctuated equilibrium. So intrinsic in this huge complexity of macro evolutionary change in DNA, because of the structure of it, intrinsic in it are two critical things. One is the vast majority of the time make a change which changes some whole network, and it's going to be a disaster. The majority of the time there's going to be stasis, and also intrinsic in that is the ability to have massive macro changes. Any of these examples we have here, where in periods of selective bottlenecks, you're going to get something like that happening. So Gould and friends are absolutely right, all of that. Not necessarily, because again, this radical rapid change period, that's for paleontologists, for biologists, that's thousands of generations in some cases. Again, what counts as very rapid in one scientific discipline may not be the same for the biologist at the other end. Nonetheless, this winds up being a mechanism for long periods of stasis and very rapid change. Okay, so that suggests that where we have these mechanisms. So how does one begin to resolve the microevolutionary picture of gradualist social biology from last week and the macro picture of punctuated equilibrium drama and all of that? How do you begin to put the pieces together? Okay, a number of ways that it could be done. Let me just see here. Yes, a number of ways it could be done. One is thinking again about micromutational changes are about changes in the function of pre-existing proteins. Macromutational changes are about the invention of new proteins, new networks, new if-then clauses, all of that serving different functions. Back to the difference in the genome between humans and chimps. When you look at the micro versus the macro differences, for example, in the domain of the immune system, most of the differences between humans and chimps genetically have to do with microevolutionary differences. And that comes down to clinical pictures that you see. Humans are much more resistant to tuberculosis than chimps are. Chimps are much more resistant to malaria than humans are. Nonetheless, it's the basic nuts and bolts of the immune system there. One of these guys has a little bit better of this, one has worse of that. This is within a realm of gradualist change. The genetic differences that explain that are microevolutionary ones. Where are the macroevolutionary differences between humans and chimp genomes? Those are the ones that have to do with development. And those are the ones that amplify differences big time. That one tiny little difference, and you get an organism that's going to look as different from a chimp as a human does the other way around. Some systems, their evolution is much more built, driven by micro changes, some much more than macro. Some of the most interesting macro stuff is going to be developmental blueprints, developmental trajectories. Okay, what else? What else in terms of resolution? There are plenty of fossil histories by now for some line that are complete enough that it is very clear they look like this. And the majority of fossil pedigrees that are that detailed enough support a punctuated model. Nonetheless, there are plenty that are understood with as many time points in here that follow gradualist models. So, evidence for both, nonetheless, mostly punctuated evidence. The biggest problem, though, in debating all of this is it is impossible, for the most part, to see gradualism going on because it's gradual, because it's hard to spot that. It's hard to show whether changes are incremental or changes are rapid, but by now there are a few examples where people have actually been able to observe evolutionary changes in organisms, and ones that can count as fairly rapid. One first example. For reasons I cannot begin to understand, people in Chicago, I don't know if it's in the Chicago Field Museum or the mayor's office or something, have all sorts of carcasses around from rats killed in Chicago in the 1880s. key to the city, souvenirs, who knows what, Chicago Centennial, but those are around stored someplace, and a few years ago researchers were able to look at the genomes of those beasts compared to rats that are being taken these days off the streets of Chicago, current ones, and showing over the course of a century a lot of the genome has evolved. There are a lot of differences. Over the course of a century, I don't know, what is that, maybe 500, 1,000 generations or so, there has been significant change over that time. Darwin's finches, for those of you Darwin history fans, that started the whole thing. All sorts of different species of finches found in the Galapagos Islands, and people have been studying them for half a century, 3/4 of a century. Gene distributions are changing. Gene distributions for traits, like the size of your bill, and thus what kind of food you could break into in response to environmental shifting there, that is being documented, that's going on. What else? Another one that is really interesting and important and critical for lots of us, which is the evolution of resistance to diabetes. This is a very interesting phenomenon. Diabetes comes in two forms: juvenile onset diabetes, that's the one where you need insulin. The diabetes that's instead becoming an epidemic is adult onset diabetes, which is when you have a body which is all built out of like lean hominid history to store away lots of calories and all of that, and you throw in a westernized diet and you get the increase in westernized problem of obesity, and you then wind up getting diabetes. For our purposes right now, it doesn't matter what diabetes looks like. The main thing is that it's driven by food excesses in westernized diets. So there's been a whole literature by now of people studying populations that have had rapid shifts from traditional diets to westernized diets. A lot of Pacific Islanders have been studied, all sorts of other populations. The group that is like the iconic one to study is Native American group in Arizona called the Kima Indians. And what's very convenient about them is about half of them live in the United States and about half in Mexico. And there has been very rapid changes in diet in the American side, less so in the Mexican one, far more traditional. And one of the things that soon emerged once westernized, sort of typical processed food became the predominant food eaten by people in the Arizona side is like 90% diabetes rates by the time you're 30 years old. The exact same thing in some of the Pacific Islands that have been studied, in Nauru and Samoa and some of these other places, astronomically high rates of diabetes. Another population where it's been studied are populations of Jews who were living in Yemen and who, I think in the 60s or 70s, moved to Israel, switching from a very traditional diet to a very westernized one. Diabetes rates absolutely through the roof. What that's doing very quickly was killing off all sorts of folks very early in their reproductive life history periods. Suddenly you got the people who are most vulnerable to diabetes when suddenly given a westernized diet, they're leaving fewer copies of their genes. And it's been in the last decade or so, some of the Pacific Island populations that have been studied, the diabetes rates are beginning to go down. In other words, there has been the selection that went into Western European populations, I don't know, a century ago or so, which is the folks who have the most efficient metabolisms and can store away all of those dozens of Hershey bars effortlessly. They're dead, and they died before they left copies of their genes. You are now selecting for human populations that have sloppier metabolisms, and this is within the course of a century. Another example, here's a totally cool, irresistible. This is one of the great genetic stories. Okay, as you can see, this pure white screen tells you that this is research that went on in Siberia. Okay, actually these were Siberia. Okay, so this was a famous study done by this Soviet geneticist starting, I don't know, about 50 years or so, where you have these Siberian silver foxes, and that's what they look like. And for all sorts of reasons, who knows what, it is highly valued to have clothing made out of their bodies, and thus highly valuable wild animals, tough to get. And this guy decided, well, what we need to do is start some Siberian silver fox domestication farming stuff. So he did a very logical thing. He started selectively breeding them for tameness. He would have a bunch of captive foxes that were wild-born, and most of them totally feral and crazed, and he would see, is there a subset, 5%, 10% of them, that I could get relatively closer to, that were calmer in the human's proximity, only let those ones breed. Now take their offspring and only take the 10% or so that are most calm, most easily tamed, and it took about 35 generations or so to get foxes that were as tame as dogs are. Something else totally amazing. So he's breeding them for only one trait, a behavioral trait, which is the ability to get close to them if you're a human, calmness on their part, whatever you might describe it as, breeding entirely based on that trait. And when you spend 35 generations breeding a wild animal like a fox so that it's the ones who are most able to function in the role of domesticated animals, it's not just the behavior that changes. This is what the adults look like 35 generations later. Oh, give me one, give me 100. I want, yes, give me, they're adorable. Can you believe how great they look? What's going on here? They were selecting for a behavioral trait, and they are fox puppies. They've got big roundy ears and these cute little short muzzle things, and they wag their tails when humans show up. And these are not dogs sent to the wolves. This was 35 generations to turn something on the left into something looking at the right. Very interesting thing we will come to in the ethology lectures. Basically, what you're breeding for when you're breeding a wild animal to be tameable, you're breeding for the ones that behave more childishly, the ones who are acting more like developing animals who are more dependent on other individuals. You're breeding them for infantile traits. This is what a baby fox looks like. This is what these domesticated adult foxes look like. Totally amazing that this happened. Ironic ending department, you will notice that the coat has changed from the ones on the left to these wonderful coats that you just want to have made into underwear and hats and all of that sort of thing. And on the right, they look like, you know, Spot and Rover and all those guys. It is called a piebald coloration pattern. Somehow also, you can't get the domestication without that happening. Ironic ending, nobody wants to wear clothes that look like that. They would have been killing Dalmatians for years if that were desirable. In the process of breeding them, they became useless economically. Nonetheless, totally amazing demonstration. 35 generations, and you turn something on the left into something looking like that on the right, who will wag its tail at you and lick your face and bring your chewed up slippers back to you. This is very rapid change. Final example. This is one that is also quite consequential, and one possibly even more so than diabetes, which is if this type of evolution keeps occurring at the rate that it's happening, all of us are going to have dramatically shortened life expectancies, antibiotic resistance, and various bacteria, evolution going on there. What we see in all sorts of cases is evolution is not just occurring over the course of glacial lengths of time, it can be occurring very rapidly. And what we see here also is, in some cases, it's going to leave a different sort of skeleton, changing how good your kidneys and pancreas is at dealing with westernized food. That's not going to make for a different fossil. Breeding for antibiotic resistance and bacteria is not going to make for a different fossil. Actually, can bacteria even make fossils? Does anybody know? Yes, okay. So it's going to make bacterial fossils that look exactly. All of this interesting, amazing stuff going on, and it's stuff that no punctuated, radical, dogmatic paleontology type would ever be able to pick up. Okay, so what are we heading to here? We are heading to a wonderful, heartwarming resolution of these different schools of thought, which is, can't we all get along, and can't we all have means of having the different types of evolution happening? So almost certainly harking back to what mentioned before, microevolutionary changes, the immune system in chimps versus humans. Macroevolutionary changes, development, you don't have to choose vanilla or chocolate. They could be going on at the same time, and they could be going on at the same time in a very important resolving way, which is, okay, so you've got evidence for a punctuated change of some trait. And single traits don't evolve at a time. They come in whole packages, packages making you wag your tail and such. So there's some other punctuated thing going on, and that's its pattern happening there. And then meanwhile, you've got some other trait moving, evolving in a punctuated manner, and it doesn't go down, but it's looking like, and you put enough of these together and have enough punctuated events happening, and on a whole population level, you've just invented gradualism. You don't have to choose, you can have it all, you can have it all. So a resolution there, both are almost certainly happening. Okay, so what does this set us up for now? Now that we have finished this bucket, we are now ready on Friday? What is today? Wednesday. We are now ready on Friday to move to the next bucket, which is a very different world of trying to make sense of the genetics of behavior. Last week's version, you make up stories built around individual selection, kin selection, group selection, etc., and come up with the best story around, show me something more predictive, and you win. This week's version showing actual genetic changes over the course of time, what patterns of evolutionary change will those code for, with what consequences for understanding the evolution of behavior, competition, reproductive strategies, all of that. By the end of this week, switching over instead to the world of behavior geneticists, where they try to understand what's going on by looking at, so somebody gets adopted. Do they share a trait more with their adoptive parents or their biological parents? You look at identical twins versus non-identical ones. You look at identical twins who were adopted into different families right at birth, put them back together, a whole world of trying to make sense that way. And what we'll see is the same deal as with this. What counts as finishing, coming up with an explanation, last week's sociobiological approaches, this version of this is the starting point for the next discipline. Okay, so we pick up on that on Friday. Oh my God, he let us out.
For more, please visit us at stanford.edu. Stanford University.
Okay, let's get going. Let's see, first off, apologies for Friday. Sometimes what seems like a flawless set of connecting flights on paper turned out not to be in reality, so hopefully people made good use of the time. Okay.
Stanford University.
Okay, let's get going. Let's see, first off, apologies for Friday. Sometimes what seems like a flawless set of connecting flights on paper turned out not to be in reality, so hopefully people made good use of the time. Okay, we are ready for our next bucket, our third one in this series, just in time to convince you that we just keep jumping these as soon as you get accustomed to one approach. Here we are, yet another one where we are going to trash everything that's come before us. Okay, what have been the two broad approaches so far? The first one, the sociobiological / evolutionary psychology one. Behavior evolves exactly under the same sort of wind tunnel of selection as does the heart of a giraffe, blah, blah, that whole song and dance, following certain rules. You could generate fairly structured predictions about social behavior. And then the we win version of using that is, here's what we predict, here is the way we explain this complex system of social behavior, using these rules which assume certain degrees of heritability of behavior, following certain rules of evolution, and until you come up with a better explanation for how this goes on, we win, this is how behavior works. Then we shifted over to the molecular end, and what we saw was, on a certain sort of trendy level, molecular biology is the answer to the people who would sit around to the social biologists and say, show me the genes for what you keep talking about inferentially. And what we saw was how evolutionary change works out in the DNA level, very importantly, the common mechanisms for microevolutionary change and how that supports the picture of gradualism, and then seeing all of these relatively new, unexpected ways in which you were getting big-time changes in DNA, all these amplifying effects of macroevolution, and suddenly support for punctuated equilibrium in two ways. First one being, as soon as you get one little tiny mutation affecting transcription factors, affecting splicing enzymes, affecting entire networks, it is going to be real rare that you don't get a mutation that's a disaster. Stabilizing, stasis, that's the long-term feature of the punctuated equilibrium. The equilibrium part and the punctuated, seeing how under circumstances of extreme selection, something that is come up with is wildly adaptive, wildly advantageous, quickly fixates in the society, and thus we have step functions. Okay, so what we do today and Wednesday is now shift to another field, thinking about behavior in a genetic context, in a heritable context, this whole field of behavior genetics. And what you'll see is, depending on your stripe, either these are a bunch of really powerful approaches for being able to infer some sort of genetic components to behavior, often getting a whole lot of insight into what's going on, and in some cases, a more negative view, a more critical one, the entire field is gibberish. And a way to summarize the view of it as being gibberish was this great cartoon I saw a while back. Okay, two scientists are standing around the lab, and they've got their lab coats on and their test tubes and going about doing science stuff, and one of them is saying to the other one, you know how sometimes you're on the phone with someone and you've been talking for a long time, and it seems like they decide they want to get off, but they don't want to say they want to get off, so they say, I probably shouldn't keep you any longer, even though you're not the one wanting to get off because they're the one who wants to get off. Well, I think I found the gene for that. And that winds up being one of the criticisms of the behavior genetics. Once again, this world of inferring genetic bases to behavior in often the most deterministic possible way, and using techniques which are often complete nonsense. What we'll see are the tools of behavior geneticists and all the criticisms and why this winds up being a very contentious field. Okay, so this being another version of getting at how do you know when a behavior has a genetic component. And we're immediately allowing ourselves to be a little more subtle here, not determined by genes, not determined purely by nature, all of that merely to have a genetic component, a genetic influence. How do you begin to do that? And this is a world where people, the scientists, don't sit around coming up with evolutionary models. They don't sit around trying to find mutations in genes. They have a very different sort of strategy. They look for patterns of shared traits among individuals who have differing degrees of shared genes, and infer relatedness and infer genetic influences from that. Okay, what's the simplest, most mindless version of doing this? The basic initial rules centuries ago when behavior genetics started was the notion, if you see a trait that is universal in a species, obviously it's genetic, obviously it's hardwired, obviously it's instinctual, obviously that's an extremely limited approach. Yes, indeed, you will see some species like flies in which certain behavioral traits are determined by relatively small numbers of genes and are universal, this is obviously going to fall apart when you get to something more interesting than a fly. So what's much more sort of the starting point for the whole field is to say, ooh, look, here's some behavioral traits that run in families. Genes run in families, therefore we may have just found evidence for a genetic influence on these traits. And of course it takes about 1/8 of a second to come up with the retort to that, which is yes, genes run in families, but environment runs in families as well. So the standard response is to now put a little bit more strictures on it to say, okay, it's not just the genes run in families, but as we know from a few weeks ago, the more closely related you are, the more genes you share in common. Our Mendelian rule of half the gene at your first sibling, etc. Following thus the logic that if you have a behavioral trait that becomes more common, the more closely related two individuals are, now you're inferring something about genetics. And of course, the problem there is not only as you become more closely related to somebody, do you share a greater percentage of your genes, you share environment more. Obviously, you are sharing environment on the average, in most cases, much more with a sibling than with a first cousin, than with an eighth cousin. The trouble is shared genes and shared environment tend to covary in families. So that greatly weakens what was the initial classic approach to the field 70, 80 years ago. Okay, so you got to come up with something fancier, something more informative, something more subtle. And what you wind up doing then is let's control for environment. Yes, obviously the eighth cousin is living in a different world than your full sibling. Let's control for environment under circumstances where you examine relatives where they have the same environment and they differ in terms of the amount of genes they share. And what is this classic approach? Look at identical twins versus fraternal twins. Monozygotic twins from one zygote, monozygotic identical twins versus dizygotic. And the general notion there is, okay, identical twins share 100% of their genes. Fraternal twins share 50% of their genes. So if identical twins are raised in the exact same environment, and fraternal twins are raised in the exact same environment, they all have environment shared. What's the only difference then to explain any behavioral differences? If you see a trait that is shared to a greater extent in a pair of identical twins than in fraternal twins, what's the only difference? They all grow up in identical environments. It's because the identical twins have twice the amount of genes in common, 100%. You could thus infer a genetic influence on that trait. Because it's the identical environment, twins get raised absolutely the same, and the only difference is the degree of relatedness, the degree of shared genes between monozygotic and dizygotic twins. That one should take about 2/8 of a second to demolish. So the simple starting point that, oh yeah, sometimes fraternal twins, dizygotic twins are different sexes. Okay, so that complicates things. So you come back and you're a little bit more rigorous now, and you restrict your comparison to monozygotic to dizygotic to same-sex dizygotic pairs. And then you institute this rule, okay, same-sex twins, whether identical or fraternal, are raised essentially in the same environment, So if you see a greater sharing of traits among the monozygotic twins than the dizygotic, the only place that greater sharing could be attributed to is the fact that they have more genes in common. We have just identified a behavior that has a strong genetic component. So the big problem with that approach is, anyone, who's a twin in here? Whoa, that's a lot of hands. Identical twins? Dizygotic twins? Any triplets? Okay, just to extend that, armadillos always give birth to 4 identical offspring at once. Okay, So, that having been prompted, what we then move on to is the obvious problem with this entire approach. Hooray, monozygotic twins get raised in virtually the same environment. Dizygotic twins of the same gender get raised in virtually the same environment. That's not true in the slightest. There is far more differentiation of environment for dizygotic twins than for monozygotic twins. They are treated differently as pairs. Thus, if behavioral traits are more in common between monozygotic twins than between dizygotic, it could be because these guys share more genes in common and or because they share more environment. A completely flawed approach in that regard. The next complication, it turns out monozygotic twins don't always have the exact same environment. Well, yes, obviously one of them gets sneezed on at a higher rate by a kid sister or something, and that's going to produce just decades worth of consequences. But even in other domains, monozygotic twins don't get treated exactly the same in some circumstances. And here is what it looks like if I had drawn it. Okay, so you got your twins, and this is during fetal life, and there are fetuses in there. And okay, there's one, and with a little thing of hair, and there's the other. Okay, and what you have is a placenta forms. And here's the deal with monozygotic twins: if they split during the first 5 days after conception, they wind up each one with their own placenta. On the other hand, if the split occurs between days 5 and 10, there's already commitment to one placenta, which they then share. So monochorionic pregnancy or bichorionic. And what does that wind up meaning? In the monochorionic, the two fetuses share bloodstream to a greater extent than in the bichorionic circumstance. In these cases, it's separate blood flow for mom. Okay, it's still the same mom and it's still ultimately the same blood, but it's going to be subtle differences in levels of stuff in the bloodstream. With a monochorionic, the environment for these fetuses are much more similar in terms of whatever is carried in the bloodstream. Okay, great. That's a great factoid, and that's like interesting things about identical twins, but like different levels of nutrients, whether it's coming off of a blood vessel here versus there. Okay, maybe that makes a difference, but these are going to be minuscule differences, nothing interesting. As one example of it, monochorionic identical twins have more similar IQs than do bichorionic identical twins. Just one demonstration that this could make a difference. This is relevant. The IQs are more similar. And this has been one reason why marmoset monkeys, pair bonding, so you know what they're all about. Among other things, marmoset monkey mothers always give birth to twins because there's two parents around to take care of them, and it's got a completely screwy circulatory system for the twins that are unlike either of these, and thus all sorts of people have been interested in marmosets over the years, the ones who couldn't afford to buy themselves armadillos, interested in terms of looking at the differences in blood flow during fetal life there of twins. Okay, so we start off here that just because you see something more in common in monozygotic twins than dizygotic, that doesn't tell you the first thing about whether it's genes involved. They share environment much more. Then even trying to make sense of what's going on in monozygotic twins, what you've got there are fundamental potential differences in environment starting at a very important early state of life. So monochorionic versus bichorionic. What else then? How about another one where you can look at sort of a behavioral trait, looking at traits and trait differences according to some genetic trait, and where environment is the same? How about gender differences? Whoa, okay, so genes have something to do with producing what your gender is, and environment being the same. If you see differences between females and males, that's attributable to the different genetics. Okay, and you should be able to nuke that one within seconds as well, which is the notion of shared environment, the notion of identical environmental experience. To give you a sense of how subtle this is, at 1 hour of life, on the average, the level of activity, the rate of movement, the amount of movement of limbs, on the average is greater among newborn boys than newborn girls. Whoa, that's there like within an hour of getting born. That's not a whole lot of time for environment having gone on there. Maybe you're seeing a strong genetic effect. What other studies have shown is from the first moments of postnatal life, mothers are already interacting differently with baby girls than with baby boys. From the first interaction, from the very first holding of them, there's differences in how long they are held. There are differences in the proximity to the body, to the face. So, whoa, sex differences in behavior at 1 hour, sex differences in environment within mere moments after being born. So that weakens that one. And this came through in another realm. This was a study that was done in the 80s that was enormously influential by a pair of scientists at Johns Hopkins, Benbow and Stanley. And it had to do with a program that I bet a lot of you guys had something or other to do with back when, which is the Johns Hopkins Gifted Youth Program thing, which I bet all sorts of you guys qualified for at various points and got, you know, the Johns Hopkins blue ribbons taped to your forehead. And that was part of this massive study that's been going on for decades and decades of kids who are very gifted academically in a number of different realms. And Benbow and Stanley were some of the senior researchers on this. And this was a study they did when they had like 40,000 kids in their database. And they asked a very simple question, which was, well, what does IQ distribution look like in this population as a function of what sex you are? And back came a very interesting finding, which was a highly significant difference in the average IQ between girls and boys in the direction of boys having a higher IQ. Moreover, when you looked at the tail of the distribution, the highest IQ range, what you saw there was approximately a 13-to-1 ratio way out at this extreme. They then followed up and did the exact same thing. No, they didn't do IQ, what they were talking about. They did math. Ignore that, erase all of that. Okay, let's start over. So lots of you probably got the Benbow and Stanley sweatshirts when you were part of that program. So they were looking at math skills. Yes, junior high school kids who were taking the math part of the SATs. That's part of getting into this Hopkins program. So they took the math SATs and they looked at those 40,000 scores and saw that there was a gender difference in the average score on the math SATs, with boys scoring higher. Not only that, but when they looked out at the tail here, okay, I'm back on track. When they looked out at the tail here of the highest math achievement, there was a ratio of 13-to-1 boys to girls. Okay, so that was pretty striking. And in their paper, which was published in Science, was a very critical phrase. Their rationale for doing the study on kids that age was the fact that, at least at the time in most schools, middle school, junior high school, kids have not differentiated classes yet. They're not yet at the point where you could choose to take extra math classes, to choose to take the absolute minimum. This was still the stage where most kids in this country are getting the exact same math classes. Everyone's still getting the same. And thus, at that point, they could include in their paper a phrase along the lines of, since all the children had essentially identical educational environments, any gender differences seen in it are reflecting, and they use the word biological, are reflecting biological differences. This was one hell of a famous study. This was front page all over the place. This wound up being shown very large article in Time and in Newsweek. The Reader's Digest, which I know sort of everyone is supposed to make fun of, but the Reader's Digest, at least, does it still exist? Yes, it does. Okay, throughout the 1980s, the Reader's Digest was the most widely read magazine in this country, which is astonishing, showing what percentage of people read in the bathroom. So having to go with the Reader's Digest there. And the Reader's Digest covered this and used the phrase, the math gene. and discussing how this was the definitive study showing that more boys have the math gene. Like, you already know that's like nonsense on so many different levels, but this was all over the place. This was the study that definitively showed genetic differences in math skills by sex, and definitively showed that this was manifest at a stage before there were different educational environments in terms of math. And what, of course, completely rips apart that study, and it was shameful that thing was ever published, let alone got as much attention as it did, is the fact that the environment was not exactly the same. Endless number of studies have shown, beginning by first grade, if it is a simple math problem at that stage for the same hands put up, a boy is more likely to be called on than a girl. Studies showing that for the same correct answer, boys in elementary school are more likely to be praised for the correct answer than are girls. By the time junior high school was coming around, guidance counselors were already differentially by sex, advising once you get into high school to take more elective math. Tremendous, massive differences in environment. And this study, using this whole argument of if there's an identical environment and you see differences, it's due to genes. And predicated on that, if that's not true an hour after life, it was ludicrous that they were making this argument about 13-year-olds. So this was a paper with enormous influence in every major newspaper in the country. Scientists have discovered the math gene, and boys have them more than girls. We will come back to that study later on because something much, much more interesting is going on. Okay, so those are the limits in the sex difference end of it. So what can you do next? There's a flip side, another approach that the behavior geneticists use, which is basically the converse. Now you look at individuals getting raised in the same environment who don't share genes. Same environment, different genes, rather than just now the different genes, same environment nonsense about gender differences, even down to monozygotic twins, all of that. But now the flip side, same environment and different genes. What was the paradigm for this? The one that is used over and over and over again, the standard approach in this part of the field, is adoption studies. You take someone who is adopted as a child, and they are now raised in a household of people who they are not related to. They're adoptive, non-biological parents. And what you now begin to look at are patterns of shared traits. Specifically, what is looked at is when you see a trait in someone who is adopted, who are they more likely to share that trait with? Their biological parents or parent, or with an adoptive parent. Now, the logic of this is completely straightforward, and this has been sort of the standard paradigm in animal studies of the genetics of behavior for centuries, something called cross-fostering. You take a newborn litter and another newborn litter, and you switch them between moms so that they're raised by different moms, and they're raised with thus someone they're not related to, but someone who they now have environmental shared with them. Or in the litter, the cross-fostering studies, you look at half of a litter and switch it to another mother, half, so you see how the iterations there go. So that was essentially the rationale for how this would be done in humans. And this was the basis of one study in the late 60s, early 70s, which was the landmark study in genetic psychiatry, in behavioral genetics as a whole, a study carried out by a guy at Harvard named Seymour Ketty. And this was a phenomenal study and phenomenally important. Here's what Ketty did. Ketty was dealing with the notion at the time of making sense of schizophrenia. And as we will see when we get to the schizophrenia lecture, the number of nutty ideas out there as to what the cause of this disease is just staggering. But what he was interested in was getting at the notion that was kind of floating around in some corners of the field at the time, which is schizophrenia has a biological component, a genetic component. And what that was viewed as very unlikely. But what Ketty did was try to go and test this. So what's he going to do? He's going to look at adopted individuals who are schizophrenic and see, are they more likely to share that trait with a biological parent or adoptive parent? You see the logic already. Okay, how many schizophrenic adoptees are you going to find out there where you also were able to figure out who the biological parents were? This was not an easy task. And Ketty's insight, his intuition, was to go to one of the places on Earth where that was most easily done, which is Scandinavia, where all the Scandinavian countries keep records like you cannot believe about everything on Earth. People understanding, for example, how the age of puberty onset in girls have been decreasing for centuries. They're always using Scandinavian data because every single thing that could possibly be recorded has been written down and stored someplace there in moth-proof vaults. And what he was able to do in ways that no human subjects coming on Earth would approve these days is go through the entire database of adoptees in Denmark and identify all of the cases where somebody adopted had wound up with schizophrenia, and then able to go back and see who the biological parents and the adoptive parents were. This was a staggeringly large study. He and a team of psychiatrists spent years in Denmark doing this, because one of the things they did was they then did follow-ups, and they themselves interviewed all of the potentially schizophrenic individuals on there so that they would have the same diagnostic standards all across the board. As we'll see in the schizophrenia lecture, it's a very squishy diagnosis. So having the same psychiatrists interviewing every possible person, hugely important control. This is part of what took them years and years. And then they finally were able to say what the patterns of similarity were. And here's what they wound up seeing. You take any random person on the street who is schizophrenic, and you take any other random person off the street, and you ask, what are the odds that this second person shares this trait with the first person? About 1% likelihood. What's that another way of stating? In the population as a whole, there's about a 1% incidence of schizophrenia. So you start with a circumstance where the biological parents, neither of them had schizophrenia, and neither of the adoptive parents did. And what's the incidence in this population among adoptees? A 1% schizophrenia rate. That's just average people off the street. That's the usual rate across Western European populations, so 1% rate. Now, let's have the person growing up in a different sort of household. This is a person who has no biological legacy of schizophrenia, but was brought up in an adoptive household with a schizophrenic parent. And what you saw then was a 3% chance, which with this enormous sample size was a highly significant, reliable number. So in a rough kind of way, being raised in a household with an adoptive parent who is schizophrenic approximately triples your risk of a schizophrenia diagnosis. But then the really critical one, which is you were raised in a household where neither parent, adoptive parent, is schizophrenic, but you have a biological legacy among your biological parents of schizophrenia. What do you see? A 9% incidence. Approximately a 3-fold increase above that, almost a 10-fold difference now over what you see in the general population. This one number was what roared through the field. This was viewed as the clearest evidence to date for a genetic component to a psychiatric disorder. Regular old person off the street, 1% rate, have a biological parent with schizophrenia and share no environment with them because you got adopted away, and almost 10-fold higher chance of getting the disease. Then, final thing, looking at the incredibly rare people who got screwed on more different fronts than you can imagine, who had a biological parent with schizophrenia and phew, got out of there and landed in an adoptive household with a schizophrenic parent. So you get the double whammy there, and what you saw was a 17% incidence. What's interesting about that number? Okay, what this cell does, what this number does, is reflect the increased risk by having a biological legacy of schizophrenia, plus the increased risk of having an adoptive environmental legacy of schizophrenia. So let's see, what's the difference between 1 and 3? That's 2. The difference between 1 and 9 is 8. So thus, it should be about 10 percentage points. It should be about a 10% rate. What are we seeing here? A synergism. Get yourself a biological legacy and get yourself a schizophrenic household to grow up in. And it was not just adding up the 2 degrees of risk, there was a synergism, a non-additive synergism. That is an important hint for us of stuff to come. So this was this landmark study. This was phenomenally difficult to have pulled off. It got Ketty a number of Nobel Prize nominations. This was the study that showed the first definitive modern science evidence for a heritable basis to a psychiatric disorder. And this became the gold standard for how to do behavioral genetic studies. And in the aftermath of that, people began to do adoptive studies on heritability between biological and adoptive households, heritability of depression, heritability of alcoholism, heritability of criminality, and you can see heading off in all sorts of interesting directions from there. All sorts of interesting ones, and then always producing a number that would be higher in this cell of the matrix than that cell, and always producing the notion that one has just shown a strong genetic component to whatever that trait is. That trait IQ, that trait criminal behavior, that trait alcoholism, these were some very loaded studies in terms of the implications. So, what is the problem with that approach? A number of problems. The first one is that under the best of circumstances for people trying to publish papers out of this, under the cleanest of circumstances, the individuals in the study were adopted away, were taken from the biological parents a quarter of a second after being born. No postnatal shared environment whatsoever. What one knows, of course, is that's not the case with adoption, and there's varying amounts of lag time before it occurs, and they were never able to factor that into the analysis. Okay, so environment, so you got 2.5 days worth of environment with your biological parents at the beginning there. Okay, so that's a confound, and give me a break though. Just a couple of days, that's going to explain differences like these. What, of course, is also linking around, and there is the huge whopping topic of prenatal effects. Prenatal environment shared with mom. And we're about to see in a little while some absolutely astonishing realms in which prenatal environment has very long-term effects. Okay, so that's a huge confound. So how could you control for that? You see a trait that an individual shares in common with a biological parent, despite being adopted away a second after birth, all of that, and thus you can infer there's a genetic component to this trait. Uh-oh, wait a second, shared environment with a mother, that may explain some of the shared trait. How do you get by that then? The difference in the likelihood of sharing a biological trait, a trait with a biological mother versus a biological father, is the measure then of the prenatal effect. If a trait is shared 10% with a biological father and 17% with a biological mother, the 7% is attributable to prenatal effects. That was the general conclusion that the field made at that point for dealing with this irksome little problem, this pesky little thing of prenatal environment, which is going to come back big time in a few minutes. That was how they were distinguished. So, some more problems with that approach. One is one that absolutely tortures behavior geneticists the world over, one which makes them just want to have people being inbred strains where they could keep them in cages and keep track of them. The problem being that often the guy saying he's the father ain't actually the father. Oh, issues of paternity uncertainty, that sure screws up your genetic studies if you're trying to attribute stuff to someone who turns out not to be related. Very much higher rates than the people at Reader's Digest would have you believe. The rate at which the person claiming to be the father is not actually the biological father. Okay, that's a bummer. That makes things more complicated. One other big confound in the adoption approach. And this was something that was emphasized by a guy at Princeton, Leon Kamen, a psychologist there, doing a very good job of showing that adoptive family placements were non-random. When a child is adopted, you don't sit there and close your eyes and spin the globe and put your finger down at some place, and 2 minutes later, this kid born in like Shaker Heights is running around with some camel herders in Rajasthan. This is not done. It is not random placement. Instead, what is a policy in virtually every adoptive agency in this country is to try to match the kids as much as possible along a number of different domains. In other words, you are also sharing a lot of biology with the adoptive parents, and that completely screws up the analyses. Adoption is non-random how it is done. One does not just spin the globe, and instead there are very intentional attempts to try to match for certain traits which have genetic influences on them. Okay, so that's a big problem. So the adoptive approach had tons and tons of interesting findings, enormously influential, but over the years, people have realized more and more prenatal effects, paternity uncertainty, and from day one being pointed out that adoptive parents have higher than random rates of shared genes with the adoptees in most cases in this country. Okay, so what becomes the next approach? And this one wound up being the gold standard, the high watermark of how to do behavior genetics. Behavior geneticists who were able to do this sort of study, the rest of the behavior geneticists hate them because they've got the best toys out there to play with, and they've got the coolest things going, and they're always snotty because they've got the best possible circumstance, the most perfect thing you can imagine, which is identical twins separated at birth. Whoa, that must be one hell of an experiment to have pulled off. Identical twins separated at birth. It turns out every now and then a pair of identical twins are adopted very soon after birth, where each one is adopted into a different household. Perfect. Perfect. Different environments, different households, identical genes. You could not possibly ask for something better than that. People like wet their pants when the identical twins separated at birth. Paradigm burst on the scene. This was so wonderful. People wrote poems about identical twins separated at birth, sonnets. It was the best. It was so informative because look at the power of this approach. This is an incredibly powerful approach. The two individuals are raised in different environments, but they got the exact same genes. So the question becomes, where are you going to find identical twins separated at birth? And there's one obsessive geneticist, behavior geneticist, University of Minnesota, Tom Bouchard, has done an entire career not only studying these folks, but God knows how he finds these people. But he first started publishing when he had 40 pairs of identical twins separated at birth, reunited in adulthood. And that made for some incredible, bizarre, heartwarming stories of discovering your long-lost identical sibling, and produced mountains worth of data. It was a bizarre literature from day one. First one being that you would get the perfect case there, and what these guys would be reporting in the literature was totally nutty stuff. Okay, so you've got one of these pairs of identical twins, and they're born, and, you know, Wolfie winds up being raised in Uruguay by neo-Nazis, and Shmuel gets raised in Israel by his highly orthodox whatevers, and then, you know, as a result of a game show quirk of fate, they're suddenly brought back together, and there's Wolfie and Shmuel, who are identical twins, and what do they report? The most amazing thing they have in common: they both flush the toilet both before and after they go to the toilet. You think I'm being facetious. Go back to that literature when that first came out and the coverage in the press, and it would be things like Wolfie and Shmuel, they both have like a poodle named Fluffy. They both, and the flushing the toilet before and after going to the bathroom, that was one of the landmark early findings of these studies. They would find twins that would do that. They would find twins who were both married to somebody named, you know, Congolia or something, and oh my God, this is totally amazing. That was what hit the pages initially. these obscure little bizarre similarities within a backdrop of, well, what's the data actually show? And what's by now a twin registry of probably a couple of hundred sets of these twins, and this has become the cottage industry of the best behavior genetics around. What has come out of that literature is the most solid, reliable findings is about 50% heritability of IQ, about 50% heritability of where you are on the introversion-extroversion scale, and about 50% heritability for degree of aggression. That's kind of interesting. And what we will see is there's all sorts of problems with this approach as well. First one being, starting right off, back to that Kamen guy from Princeton, his critique, turns out there were not a whole lot of Wolfies and Shmuels, even though they got adopted into different families. There again was the non-random placement in families, more similar environments than one would have anticipated purely by chance. So that as a confound. Okay, so what's the solution for that one? I know, let's look at monozygotic twins separated at birth and reunited on Oprah at age 50, and then look at dizygotic twins separated at birth and reunited after the commercial break on Oprah, and see what the similarities are. And if you see more things in common with the monozygotics rather than the dizygotic, you've just controlled for the non-random placement in the adoptive homes, the extent to which the monozygotics have traits more in common than the dizygotics. That reflects the identical genes. That was the interpretation. That was a very powerful sort of analysis, one that nonetheless winds up being very limited because, in this case, because of tiny sample size. It's really hard to have done those studies. What's another feature of the whole behavior genetics approach? Here's another one. If you see traits that occur, behavioral traits, in the absence of any learning, in the absence of any environmental experience, in the absence of anything that can count as being non-genetic, if you see that, you're looking at a genetic influence. And what are the examples that are always given? The fact that all babies all over the universe start smiling, and they use the exact same set of muscles, and they always start smiling socially around roughly the same age. And what you also see, I don't want to know how these were done, but these classic photographs that you can get filming of kids in utero, I don't know what, fiber optic something or other was doing that, but the demonstration that fetuses smile during the third trimester. This is a motoric pattern that is shared among all humans. Okay, well, social smiling, you see the flaw there, which is that is very subject to shaping of behavior. You are 3 months old and you're watching all this social smiling going on around you, and how the mannequins they have at home are nowhere near as interesting as the animated faces there, and you sort of try it out yourself. What you see is the exact same developmental time course for smiling among congenitally blind babies who never see anybody smile. So you see the motor pattern here being something that, is arguably fairly universal and occurring in the absence of any sort of training how you go about smiling. The other example that's always used is with congenitally deaf babies. And what you get there is another universal, which is beginning to babble at the exact same age that hearing kids begin to babble, and the same argument being made there. Of course, you have a very uphill task there of ruling out any environmental similarities. Because once again, an area that has been utterly underappreciated in this whole field is the whole world of prenatal environmental effects. And the theme that's going to come out of that is environment does not begin at birth, and some environmental effects prenatally are enormously influential forever after. And if you showed up on the scene 1 second after that individual was born, all of the tools of modern behavior genetics would tell you that you were looking at a genetic trait where it is one instead that was brought about by the prenatal environment. Okay, let's take a 5-minute break and then we will pick up on that. Good ones. First one being, going back to that gender difference business, the assumption running through the field that, okay, boys and girls are raised in the same environment by their parents, and the only thing that differs is the genes. Like, even those people, behavior geneticists, believe that could be the case? No, they've recognized this was a very limited approach, and thus would limit themselves to circumstances like the first hour of postnatal life, which we already saw as a flawed assumption, or under circumstances where everybody's had the same number of math, problems to take in their first 12 years of life, and we see the problem with that. Nonetheless, there is a recognition that that is a very limited set of tools for getting at these issues. The other useful thing that was brought up was somebody pointing out that in the extended notes for behavior genetics, in the second paragraph, somewhere in there, about the fourth or fifth line, is the very clearly typed out word unable, and pointing out that actually the word was supposed to be able. So you might want to take a look at that and kind of keep that one in mind. Okay, perhaps I should take a look at it also. Moving on now to this business, all of the approaches we've been seeing about comparing monozygotic with dizygotic. By the way, with the identical twins, 2/3 of them have monochorionic, 1/3 are split. And how many of you who are identical twins absolutely know in your heart of heart right now whether you are a monochorionic or a bichorionic twin? Okay, well, that didn't work very well. Okay, so pushing on, what we see here is with all of these approaches, the adoption, the twins separated at birth, the twins mono versus dizygotic, et cetera, all of those were predicated on one simple assumption, which is environment begins at birth. And that has been completely destroyed in some incredibly interesting ways in recent years. We have very vibrant literature at this point. First way that it can go down, which is your prenatal environment. What are you having as a prenatal environment? Who are you sharing environment with? Obviously, with your mother. You are sharing blood. You are sharing blood, and thus the things that she is experiencing in the world around her that make for a different environment than the person standing next to her gets translated into effects on the fetus. First domain, hormonal ones. Here's one example of something that you will wind up seeing. This was work done by a guy named Fred von Saal at University of Missouri. And what he did was look at the fact that rats give birth to litters of about a dozen kids at a time. And there's some circulatory system thing. They look like a whole necklace of fetuses there. And a circulatory system is such that everybody's getting blood, but you're getting preferentially the blood that is from the siblings right around you. There's some sort of loop of the blood system that looks just like that. And what you wind up getting is you have a particularly shared blood environment with the siblings on either side of you. And what he asked was something very simple. You are a female rat fetus, and in one case, you're sitting there with brothers on each side. In another case, one brother and one sister, or in the final case, obviously with two sisters on either side. And what you wind up getting is a different hormonal environment. How does that translate out later? The more male siblings you have around you as a fetus, the later you're going to reach puberty. That's interesting, suggesting that very local endocrine effects here play out in something like that. Also, it predicts how estrogen levels are going to drop in you later in life. So this winds up being one very interesting prenatal environment. Here's another one. Here we have in humans the age of one's mother when she gave birth, and extrapolating a little bit here at both ends, but just assume this is kind of the age range. And what we see here is the age of puberty onset in the offspring. And what is seen is very young mothers and very old mothers have offspring who reach puberty later than women in a more intermediate age. What does that appear to be due to? Differing estrogen levels. Higher levels of estrogen at this point in life, it's actually not symmetrical, it's skewed a little bit this way, And that seems to be the driving force on it. Whoa, the age at which you reach puberty has to do with how old the fetal sac was that you hung out for 9 months. That has an influence, absolutely. So prenatal effects. More, another version of it. Suppose now the hormone you are getting inundated with through the bloodstream is a stress hormone. A stress hormone, glucocorticoids, we will learn all about those down the line. A stress hormone because mom is stressed. What are some of the consequences? For the same prenatal stress, as an adult, you will have a smaller brain. If you're a rat, you will have a thinner cortex. You will have less learning abilities. You will be more prone towards anxiety. You will have fewer of those benzodiazepine receptors that we heard about at the other day. You will have more of a cognitive decline when you are a doddering old rat. All sorts of stuff will go differently throughout your entire life. But get this. Okay, look at this mechanism. So you are a rat and your mother was stressed when you were a fetus back when, and you were marinated in those glucocorticoids when you were a fetus. Your brain overall will be smaller. There's one particular brain region, which I won't mention right now because it's not really critical. There's one brain region that's particularly hard hit. What does that brain region do? Among other things, it helps to turn off the stress response. So if that part of the brain is smaller, you're not as good at blocking glucocorticoid secretion at the end of stress. And somebody with a normal sized whatever, something stressful occurs and they recover, and you do this instead. Because this mysterious part of the brain is smaller, is not giving as much of a negative feedback signal. And for people new to endocrinology, that's something you'll be getting in a couple of weeks. The net result is, if this part of the brain is smaller, you will have more lifetime exposure to glucocorticoids. So what happens next? What happens next? In addition, baseline is also elevated in these individuals. So the net result is a lot more cumulative exposure. So you are a female rat and you were in a mother who was stressed prenatally when you were a fetus, and as a result, in addition to all the other problems that you've got lifelong, you secrete higher than expected glucocorticoid levels, and eventually you get pregnant. And thus, your fetus is going to be exposed to elevated glucocorticoid levels and will be born with a somewhat smaller brain, thinner cortex, etc., etc. What have we just shown? An environmental manipulation on a pregnant female manifesting itself 2 generations later in the grandchildren. And when this was first described in the early 60s, this was called the grandmother effect. And eventually it was shown to go out about 4 or 5 generations. The magnitude of the effect would get smaller with each generation before it disappeared. But look at what this is about. This is inheriting a trait that is not genetic. And this wound up being the first example of what is now called non-Mendelian inheritance of traits, non-genetic inheritance of traits. And all you've got going here is prenatal environment, extremely powerful observation. And what you also then have is you're some researcher, and again, you come along 1 second after the animal is born, and you wind up studying saying, oh, look at this rat tends to have elevated glucocorticoid levels just like mom. And this rat tends to have a thinner cortex just like mom. And this rat, and if you've never heard of prenatal environmental effects, what's the only conclusion you could make? There are genetic influences on these traits. So these non-Mendelian, non-genetic transmission of traits are really, really important. Next thing, what else is floating around in mom's bloodstream that gets shared besides hormones? Nutrients. Nutrients. And this winds up producing something extremely interesting as well. Okay, it's all a blur by now. Have we already talked about Dutch hunger winter? Oh. Okay. Well, now that you all know about, what did I say? Okay. Okay, for those of you who didn't, Dutch hunger winter, here's the deal. If you were a fetus in Holland during the winter of 1944. Something very interesting happened with you. 1944, Holland is still occupied by the Nazis. Nazis are being pressured on all fronts in the process of losing. They're falling back. They're losing a lot of the land they've occupied. And what they decide to do that winter is, because they need some food and because they wanted to punish the Dutch for beginning to be more openly in the resistance, what they did was that winter they diverted all of the food in Holland to Germany. And this historically is known as the Dutch Hunger Winter. Essentially, people went from a perfectly fine, healthy, amid the context of a war, Western European diet down to something like that from out of nowhere. 40,000 people starved to death during the Dutch Hunger Winter. Something very interesting occurred if you were a third trimester fetus during the Dutch Hunger Winter. Because it only lasted for the winter. The Allies came in, liberated Holland after that, and it went to something like this. Something resembling a step function of starvation for about 3 months. If you were a third trimester fetus during the Dutch Hunger Winter, your body learned something important, which is there's not a whole lot of calories out there. During third trimester, fetuses are in some way, and this is metaphorical, deciding, learning how much is the way of nutrients out there in the world, how readily do calories come in, how is the fetus finding out by way of mom's circulation. Mom is starving, and thus much lower levels of nutrients in the bloodstream, and the fetus at that point in development is saying, metaphorically, well, what's it like out there in this place I'm going to be heading to rather soon? What's the nutritional profile like? There's not much in the way of food out there. And as a result, the fetus has, and the term used now is metabolic programming. There is metabolic programming or metabolic imprinting. Notice the word imprinting here being used in a completely different sense than we've heard about already. Metabolic programming to produce what is called a thrifty phenotype. The fetus, realizing there's nothing out there in the way of plentiful food, what it does is it programs its pancreas to function in a certain way forever after. What does the pancreas do? The smallest smidgen of food hits the bloodstream, and the pancreas is pumping out insulin, which helps store all that stuff and quickly scarf up every bit of nutrient in the bloodstream and store it away, because you got to be as efficient as possible. And you metabolically program your kidneys so that your kidneys are incredibly good at retaining salt, because along with starvation, there's going to be salt shortages. So really hold on to salt, and you are born with a body with a thrifty metabolism. Very good at retaining salt and spectacular at storing away every bit of nutrients that hits the bloodstream. So at that point, you go back to this sort of diet, and you have that for the rest of your life. And what has now been shown with the Dutch Hunger Winter individual, the ones who were third trimester fetuses then, as adults, they have a 19-fold increased incidence of obesity, hypertension, diabetes, and what's called metabolic syndrome. What's that about? Their body has programmed to be extremely thrifty with metabolism, and as such, it is forever after storing away every bit of the grotesque westernized diets that we all wallow in. And what you've got then is setting individuals up for a much, much higher risk of these metabolic disorders. If you were a newborn at the time, didn't happen. Newborn during here, the metabolic programming is already over by then. If you were a first trimester fetus, didn't happen. The metabolic programming hasn't started yet. Second, the later part or second to third trimester is when the programming goes on. And this was a landmark observation. This was really important. Among other things, you get the people who do not like the notion of these subtle effects, and they're saying, okay, sure, this can happen, but this is subtle. 19-fold is not subtle. And this was the landmark study that ushered in what is now called a whole field of fetal origins of adult disease. Fetal origins of adult disease, which a lot of realms of medicine think is outrageous and couldn't possibly work this way, and it is popping up in more and more domains.
Elevated levels of stress hormones during fetal life, increased likelihood of anxiety disorders as an adult, independent of postnatal environment. Other examples like that, all of these being ones of programming around that time. And the Dutch hunger winter one is the iconic example. You know, what we're all accustomed to is, yeah, you study something in like a planaria, and then you study it in a rat, and you study it in a monkey, and then you study it in a college freshman. And finally, when it's, now you can conclude something maybe about humans. This was first discovered in this population of humans. This was is this relevant to humans? Interestingly, there was another population at the time that went through something or other like that, which was people in the city of Stalingrad who were under siege by the Nazis and had essentially years' worth of severe starvation. They didn't get a Dutch hunger winter phenomenon because the food posted off like this, and afterward it took years for it to reach a Western European average. You don't get it under the circumstances. It's a step function like this. Okay, so think about this now. So you were a Dutch hunger winter fetus, and as a result, you have a very thrifty metabolism, and 30 years later, you've gotten pregnant. You're having a perfectly normal diet, normal intake of calories, but you've got this thrifty metabolism, and as a result, your body is really good at pulling nutrients out of the bloodstream, because you secrete more insulin than most people would with your thrifty metabolism. So you are pulling a disproportionate share of the calories out of the bloodstream, and thus your fetus is getting a disproportionately smaller amount of calories. And thus your fetus is born with a milder version of the Dutch hunger winter phenomenon. And this has now been shown in the grandchildren of Dutch hunger winter fetuses. This is that exact same deal. This is non-Mendelian, non-genetic transmission of traits multi-generational. Absolutely astonishing that this could work, and the biology is all in place for it. What we will see in a little while is what the mechanism is, and it's been identified down to the molecular level of what went on in these Dutch hunger winter. Okay, I'll give it away. Remember that epigenetic business the other day? There are epigenetic changes in the genes coding for things related to insulin in the Dutch hunger winter babies. So, enormous effect. Another example of it, another dietary one, which is if you were female and female human, among other species this has been shown in, there's an issue of how much estrogen does your mother consume when you were a fetus? Where's estrogen coming from in a diet from a lot of different types of plants? Phytoestrogens. And what the studies show is that increased exposure to estrogen derived from phytoestrogens during fetal life, and there's a small but consistent increased risk of estrogen-dependent breast cancer. 20 years later, 90 years later, again, very subtle prenatal environmental effects playing out forever, even unto the generations after you. Another realm of prenatal effects, learning. Okay, this sounds ludicrous right off the bat that you can have learning prenatally. You have learning prenatally. You can show this first in rats. Here's what you do. You take a rat fetus and you can inject into it a particular flavor of water along with sucrose. And the fetus absorbs it. The fetus actually drinks amniotic fluid, which I find to be deeply creepy. But nonetheless, you inject this stuff in there, and you were doing this a number of days running, and this fetus is now drinking this flavor that has a lot of sucrose in it. It tastes good. It tastes good. You're a fetus. What do you mean it tastes good? The fetus learns about it because after birth, given a choice between two neutral flavors, it will prefer the flavor it was exposed to that it was drinking when it was a fetus. How weird is that? More ones. Okay, that was good grammar. More ones. Here's one from humans. And this was a study which is as strange as you can get, looking at the fact that the diaphragm is a very good resonating membrane, something or other. Mothers' voices are heard in the fetal sac quite readily. And if I don't know how they got the fiber optic camera in there, I have no idea how they got the microphones in there for recording that, but it is a very resonant chamber, the diaphragm and the amniotic fluid. Fetuses hear a lot of what's going on out there, most notably mom's voice. So this was a study, a totally inspired one, and what you had was, in this one, pregnant women spent their last trimester loudly reading either The Cat in the Hat over and over, like 4 times a day or something, until they went mad, absolutely mad, and that's before the kid was even born, or reading some random collection of sentences that control for word length, that control for rhythmicity, all of that. Then you get the newborn sometime later and you give them a test of which they prefer to listen to. How do you do that with a newborn? Something you can do is when newborns like something, they make more sucking motions with their mouth. So you've got a suckometer thing in there measuring it, and the newborns prefer hearing The Cat in the Hat. They learned it. Not a huge effect, but nonetheless, what is up with that? Of course, a follow-up question that maybe half the people in here would be wondering is, well, what about the fathers? And they had the study with the fathers reading Cat in the Hat and, you know, like a megaphone on mom's belly and reading, and doesn't work, doesn't resonate enough that side, sounds from the mother. Okay, so what we see here is this whole world of stuff going on before behavior geneticists show up and say environment has just started. All these prenatal effects of hormones, nutrition, sensory stimulation, amazing, in some cases multi-generational. Yeah. Where are they now? Remarkably, all of them are heads of states of countries. People are still trying to understand that one, but very good follow-ups on that. So we've got this punchline here over and over showing the power of prenatal effects. One final study, and this was one carried out by a scientist at Berkeley named Darlene Francis, which took some amazing surgical skills. So there are different strains of rats that have been bred for different levels of anxiety. And we've already heard about one possibility for that. It turns out some genetic differences in the promoter to the gene for the benzodiazepine receptor. There's all sorts of strains that have been bred for high, low levels of anxiety that people have studied. They've been bred. These are transmissible traits. These are heritable. These are genetic traits. Here's what Darlene Francis did, which was she did an adoption study. What's the adoption paradigm we've heard already? Right after birth, you cross-foster the rats or the kids get adopted. That's not the adoption study she did. She transferred fetuses. She figured out how to do the surgery to move a fetus early on in development from one rat mom to another rat mom, where they develop perfectly normally once she had the surgery down. And you know what turns out to be the case? It's not a genetic trait. It was not a genetic trait. You grew up with the anxiety levels matching the strain of your mother, even if the strain of the individual whose body you fetally developed in. You take a mom from the high anxiety strain, and you take a mom from the low anxiety strain, and you take fetuses from the high anxiety mom, and they go through gestation with the low anxiety mom, and as adults, they are low anxiety. It was not a genetic trait, it was a prenatal one having to do with another one of those multi-generational begatting by having early experience influencing the nature of a pregnancy you would eventually have, and thus influencing your fetus, producing a different pregnancy for them down the line. This demonstration of cross-fostering, of adopting as early as you possibly could. Enormously important study, one really difficult one, yet another way of pounding on this point, environment is not taking at birth. And some of the most important environment is not occurring starting at birth, and everything about behavior genetics classically was predicated on there's no environment before that. Okay, so what do they come back with? What's the response? We've already seen a possible way of controlling for that, which is if you see traits shared in common with the biological father, and then you see more traits shared in common with the biological mother, the increased degree of sharedness with the mom reflects not the genes, because you're getting the same genes from the same amount of genes from each parent. It reflects the prenatal environment. That would be the control that would be used in this study. The extent to which the trait is more shared with a biological mother than with a biological father is a reflection of prenatal effects, because you get the same amount of genes from each parent. Naturally, this turns out to be vastly messier than this, because there's a whole world in which you're getting more genetic influences from your mother than from your father. Okay, first one is, once again, if it turns out the person who's claiming to be the father isn't actually the father, that changes the whole map. And once again, that is a problem running through all of human behavior genetics literature. But here's the next one. Here we have, just to make sure we've got our cliche in place here. What is this? This is the, okay, which is the what of the cell? The powerhouse. Okay, here we have the powerhouse of the cell, mitochondria. And in this rather odd cell, there's one mitochondrion, but it is standing in for all of the powerhouses. And we've got mitochondria. Something that when you first learn about this is just flabbergasting, which is here you got a cell with its DNA and its nucleus and a double helix thingy happening there. And it turns out that mitochondria have their own DNA. And this is part of explaining one of the truly amazing, adventurous, nutty ideas that have turned out to be true. Scientist named Lynn Margulies, University of Massachusetts, 30 years ago or so, she noted that this business of mitochondria having their own DNA and came up with this hypothesis that mitochondria used to be independent organisms that in some symbiotic whatever billions of years ago got into cells that had no mitochondria at the time, and that there's been a symbiosis ever since. Mitochondria have DNA which are related to mitochondrial function. Not a ton of this stuff. Nonetheless, you have every gene that is critical for mitochondrial function. No, that's not true. A few of them are wound up in here, but all of the genes in here are pretty important for mitochondria powerhousing, all that sort of thing. And these are derived from a completely different world of DNA than these. So now consider this. This cell is an egg. This cell is not an egg. This cell is just merely carrying genetic information. That looks like that. What you've got is sperm. All they're carrying are the DNAs, the genes, the DNA. And they don't have cytoplasm, the fluidy environment in a cell. This is one major dense packing job. Here's the critical implication. Eggs have mitochondria. Sperm don't. So right at the point of fertilization, you have gotten all of your mitochondria from your mother. And thus, all of the genes related to mitochondrial function that are contained in the mitochondria, you don't get from fathers. It is exclusively inherited from mothers. Mitochondrial DNA solely comes from the mother. So what we've just seen as a first pass is it's not an even 50-50 split. You get a disproportionate share of your DNA coming from the mother. Really important, and one that's been used by all sorts of evolutionary geneticists to trace legacies. If this mitochondrial DNA is only passed along female lines, that allows you to figure out all sorts of stuff about evolution. It has given rise to the Eve hypothesis that somewhere back then was a woman, probably some sort of early hominid, who is ultimately the ancestor, the great-great-great-great-great-grandmother of every single human on Earth, and it can be traced through the mitochondrial DNA. So that's an asymmetry. Next source of asymmetry is back to that business about imprinted genes. from a couple of weeks ago, you remember that one, genes that are working differently depending on which parent you are getting them from. So that one's a violation as well of this rule that you get all of your DNA in equal amounts from each parent. This is another thing that possibly skews the ratio. Now here's another very interesting thing. So you got the egg here, and not only does it have this cytoplasm with mitochondria floating around, but in addition, there's other stuff floating around in there, like transcription factors. Sperm don't have transcription factors. Sperm, all they're doing is on this like suicide swimming mission there, and they're not making any new genes. All they're doing is this one long spurt there of racing for the end. And in the case of the eggs, though, you have transcription factors. You have all sorts of proteins in the cytoplasm. You've got a fully functioning cell instead of this sort of much more streamlined version. All the transcription factors that come in a fertilized egg are coming from the mother. So what does that wind up meaning? Transcription factors, those are proteins. We're not talking about genes here. We just saw how you get more genes from your mother than your father, these mitochondrial genes. Okay, but transcription factors, the father has genes for transcription factors, the mother has genes for transcription factors. What's the significance of getting your transcription factors from your mother in the fertilized egg? Okay, consider here two genes. The first one codes for transcription factor A. We know that already. Transcription factors are typically proteins, so they have their own genes all the way down. And this codes for gene X, whatever that one is. So here's what you got. There are promoters responsive to transcription factor A. Transcription factor A turns on the synthesis of the protein coded for by gene X. And in addition, transcription factor A turns on transcription factor A gene. It's a positive feedback loop where it makes more and more of the stuff. That's the way this particular transcription factor works. So suppose this is the only thing that can activate transcription of gene X. So suppose you've got some environmental event which as a result of it knocks out the activity of transcription factor X in an egg. The egg is fertilized, and as a result of transcription factor A not being expressed, it doesn't express more. This is the only thing that drives more expression, and you never make gene X. Now, somewhere along the line in your body, you are soon making eggs which contain these genes, of course, but where you have never expressed transcription factor A, because this was knocked out in the egg, so because of that, you never express gene X. And that egg gets fertilized, and you pass on that trait to your offspring. You pass that on, this acquired trait of transcription factor A not working. If this is the circuitry that you have, it doesn't matter which version of gene X you get, you are never going to express that gene for generations and generations forever, if this is the loop. What's going on here, what a lot of people think is relevant, are some environmental toxins that are known to disrupt the activity of certain transcription factors, and what that does is induce heritability in a non-genetic way of non-expressing the gene. The gene's being inherited, but it will never ever be expressed. What have you just acquired? A Lamarckian trait. You remember Lamarck. Everybody learns about Lamarck in order to mock him viciously. And Lamarck had the notion that the way evolutionary change works, the way inheritance works, is you experience something and it causes a change in your body. And as a result, you pass on that acquired trait to your offspring. Ludicrous Lamarckians have been mocked and pilloried for centuries, except in the Soviet Union in the 1930s, where it gave rise to Lysenkoism, a very horrific piece of genetic history. But what you've got is this complete trashing of the notion of you acquire some trait from the environment and you pass it on to your offspring. This is Lamarckian inheritance. This is an environmental factor that knocks this transcription factor out of action, And if this is the wiring that you've got, this gene will never be expressed, and as a result, it will never be expressed in your offspring and your grandkids, etc., all the way down. This is Lamarckian inheritance of the trait. And again, where the best evidence for this has been is with environmental toxins that knock out, that have some of these mutating effects in eggs that are not mutations in a classical DNA sense, but nonetheless they are now heritable. So that pops up also. So what have we got here? We have the simple assumption that if you see more sharing of a trait with the mother than with the father, that's reflecting prenatal environment. And what we've seen here is totally messing this up is the fact that you do not get equal genetic influences from each other. You are getting more genetic material, you are getting more genes from your mother because the mitochondrial DNA. Even if you're getting equal amounts of DNA, expression of them will have different consequences because of imprinted genes. Finally, in this world having nothing to do with the amount of genes or the actual DNA, where you can have this Lamarckian inheritance of traits due to environmental perturbations, what we see here are ways ranging from extremely subtle and rare to some rather substantial ones with the mitochondria, where you are not getting equivalent inheritance from both parents. So that confuses things a lot. Okay, so after all of that, you do nonetheless get circumstances where behavior is influenced by genes. And by every rule, it could be shown, and by the most contemporary of techniques where people find the gene and the DNA and they've traced out the steps and showing that there really are genetic influences on behavior and ones that withstand every single one of these criticisms. Okay, so sometimes you've got genes regulating or genes influencing behavior. But now we bring in a whole other possibility. And this is something that was emphasized by a psychologist named Judith Rich Harris a number of years ago, a very important book of hers called The Nurture Assumption, which has a lot to do with arguing the relative importance of influences of peer versus parents. Nonetheless, she focused in one section of the book on the genetics of behavior and focused on what she calls indirect genetic effects. What would indirect genetic effects be? Okay, you've got that trait that I referred to before, one of the most reliable of traits in the identical twin separated at birth business, 50% heritability of where you are on the introversion-extroversion continuum. Okay, so that one is held up pretty well amid all the possible sort of complaints about these various approaches. That one appears to be quite solid, and you're immediately off and running with, okay, genes for extroversion, for sociality, for all of that. What she shows instead is something else is happening. There's a very, very heritable trait from parent to offspring, one of the most heritable physical traits out there, which is your height and your appearance in general. That those are highly heritable traits. And suddenly you have a phenomenon that is well documented, which is people who are taller are treated better and considered more attractive, comma, he says bitterly. What you've got is people are treated differently along those lines, and what is known also is people who are treated more positively during development, during childhood, become more extroverted. What we have here is not heritability of the trait where you are in the introversion-extroversion continuum. What you have is heritability of a physical trait which causes you to be treated differently in the world, which then brings about changes in personality. And studies have since shown that most of the heritability of the introversion-extroversion is mediated by physical traits in between. So that's a completely indirect way in which you could have gotten to this. More cases of this. Let's see. Okay, you can show in various bird, turkey, hen species that there are chicks that there is heritability of rank. You could be born to a low-ranking mom in the pecking order, high-ranking, all the perfect studies and controls and cross-fostering, all of that, and there is heritability of rank. But another indirect genetic effect that was subsequently demonstrated, which is it's not the rank that's being inherited, it's a particular version of genes related to melanism of your feathers, the color and iridescence of your feathers. And it turns out if you have a certain color pattern, all the other like roosters and chicks and hens and poultry peck at you more often, and you're reduced to subordination. This is not inheritance of a social dominance trait or a social subordination trait. This is inheritance of a gene having to do with the color and iridescence of your feathers, which wind up producing your social rank. Another example. Here's another one, okay, back to chicks again, which is chicks appear to be intuitively able to peck at grubs shortly after birth. That they're able to peck down and get grubs, and by all the rules of behavior genetics, with all the constraints and criticisms answered, this appears to be a heritable trait. But it turns out that this is not what is heritable. What is heritable, bizarrely, is the tendency of newborn chicks to find their toes to be very interesting and to peck at their toes. And they quickly learn that this doesn't feel all that great, but if you do it in somewhat more sloppy of a way, you get one of these things that's squirmy that tastes good. They start off, what is genetic is the tendency to peck at your feet. God knows why. If you were a newborn chick, because of extremely elegant high technology studies in which newborn chicks are put in galoshes or something, and they don't peck and they don't show the seemingly innate ability to peck for grubs. So here we have a behavioral trait which in fact is indirectly mediated by something else. More examples. There is by now a literature showing approximately 70% heritability, and I keep using this word. We are going to dissect the word heritability big time in a short while. There's about 70% heritability of political party affiliation in this country. Sharing that behavioral trait with your parents. Whoa, what is that about? That's sure disturbing, and that sure makes you want to go procreate in the name of your political stances or whatever. And this appears to hold up pretty well to some of the standard criticisms in the literature, and it's got nothing to do with this. What's the mediating variable? A large, really interesting literature showing when you compare political or social progressives with political or social conservatives, one of the most reliable personality differences is how they feel about ambiguity. Conservatives, on the average, do not like ambiguity. They are much more ambiguity averse, and you can start it with showing ambiguous sensory stimuli in kids and looking at heart rate responses to it, and that tends to be a stable personality difference at political extremes. We will see sometime later on that there's a whole world of moral development in kids where there's various scales measuring at one called one's Kohlberg stage of moral development, that there was an old literature suggesting political differences as to how fancy of a Kohlberg stage you got to. We will see that despite what struck me as the intuitive sort of logic of what was found, that one hasn't held up. But one that does hold up is difference in ambiguity tolerance. And that's probably the mediating trait. It is not inheriting a tendency to like elephants or donkeys or whatever. It is instead having this intermediate trait. Final example of what would be an intermediate trait. There's a whole bunch of rat and mouse strains that have been developed that have differing levels of aggression. High aggression strains, and you do all the proper controls, and you can show this is a genetic trait. Whoa, heritability of aggression. We're suddenly back to twin adoption studies in Ketty with heritability of criminality, all of that. Heritability of aggression, what's actually going on in all of the strains identified to date by spontaneous traits coming up and then breeding for it. What you see instead is the strain that is so aggressive and so pissy and so impossible and so unconstrained by the law and order of rodent society and all of that. They've got lower threshold for pain sensitivity. Things hurt them more readily, and they're more likely to become aggressive at that point. It turns out it's genetic differences in the neurobiology of pain sensitivity. So what we're seeing here over and over is, amid the gazillion of criticisms we've had about when does environment actually start and when do environmental assumptions of being treated the same go down the tubes and differential inheritance of genes. Yeah, some traits do appear to have some fairly strong genetic components, but even once you get that far, they very often are through some very indirect routes. Okay. More things here. More things, we just covered that. Okay, a little bit more in epigenetics, which is that whole business you remember from the other day, which is, and you got the DNA, and then you have that whole world of protein coatings, which I've been very careful not to give the jargony name for because it's not important. But what you've got is this whole world where regulation is not so much at the level of genes and the 95% of the DNA that's the on-off switches, but whether the transcription factors can even get in there. And this world of epigenetic changes that will cause lifelong differences in how readily transcription factors get to something or other. What have you got at that point? The exact same possibilities as this one here. If instead of, due to some environmental toxin, you knock a transcription factor out of business in a fertilized egg where there's this setup of genes, if for some reason, whatever, you do not have access to it because of an epigenetic change, it's going to be the exact same consequence. Multi-generational inheritance of non-genetic traits due to epigenetic rather than genetic differences. That is turning out to be what went on in the Dutch hunger winter people and the animal models of epigenetic differences in access of transcription factors to genes related to insulin metabolism. That turns out to be a critical one. Here is one of the coolest examples of this to date. And this is work done by a guy at McGill University named Michael Meany. And what he is focused on is what started off as a very artificial literature, which is take yourself a newborn rat, and for the first 2 weeks or so of its life, every day you pick it up for 3 minutes and you pet it. And now you put it back. And all else being equal, it will have a bigger brain in adulthood, better learning abilities, more resistance to a whole bunch of neurological insults, lower glucocorticoid levels, etc. That whole world of what came to be known as neonatal handling. On the other hand, pick up the rat, take it away from mom for, instead of 3 minutes, an hour and a half, then each day put him back, and as an adult it's going to have a smaller brain and a shorter life expectancy. 3 minutes away from mom does wonders, an hour and a half of being petted does not. That in and of itself is interesting in terms of what counts as stimulation, what counts as stress. Okay, so hooray, what we've just learned is just how generations of rat petting graduate students can influence the lineages of rat brains and all of that. And what Meany started looking at, with this phenomenon being one that was around forever, first identified around 1960 by a guy named Seymour Levine in the psychiatry department here, no longer alive, but that started this whole world of neonatal handling. What Meany did was say, well, rats did not evolve whatever's going on here for the purpose of doctoral theses. What's the natural equivalent in the world of a rodent? And it turns out that what happens when you pick up a rat for 3 minutes and do this and put it back, mom is all excited and goes and checks out the pup and nuzzles it and nestles it and licks it and whatever other stuff there, and has all this attention. Whereas if you take the pup out for an hour and a half and you put him back with mom basically ignores the pup for long periods of time. You're changing the mother's behavior. So that's an indirect effect. And what he proceeded to show was the critical thing about handling is not what you're doing to the rat during that time, it's the fact that you're causing dramatic changes in maternal behavior based on that. So that's interesting. But that still doesn't solve the problem of why did this system evolve for grad students manipulating maternal behavior. And what he then proceeded to look at was normal variation in rat mothering styles. Because some rat mothers are, okay, I know this is a value judgment, but some rat mothers are better mothers than other mothers. Some rat mothers just simply are better. They're better, they're nicer, they have better souls. And in these rat mothers, how do you determine that by these sorts of measures? Licking and grooming. How much time do you spend licking your baby? And how much time do you spend grooming your baby? And what Meany proceeded to show is that's what the neonatal handling phenomenon was about. When you have moms who lick and groom their kids an awful lot, what you do is produce the same sort of better outcome from the 3 minutes of petting deal there. You get the kid who is bigger and healthier and smarter, that sort of thing. Moms who hardly ever lick and groom their pups, they produce pups that as adults are like the ones that were separated for an hour and a half a day. It is a reflection of mothering style in the rats and the variability there. Next thing he showed was that this was multi-generational. If you lick and groom your baby rat daughter a whole lot, as an adult, she will be more of a licker and groomer. And he's already shown what some of the neurological mechanisms are for that, for development. What have we got? Yet again, one of these non-Mendelian inheritance of traits deals going on. In this case, not even prenatal. Your early experience is going to cause lifelong changes in your brain, which will make you more likely to reproduce the same early experience for your offspring. Off you go. The final thing he did, which stands as a landmark in the field of behavioral neurobiology, is he figured out what the epigenetic change is. One of them, or rather two of them, is identified by now. What gets changed by how mom often or unoften licks you, grooms you, all of that, you change the access of transcription factors relevant to making genes, to activating genes for making receptors for stress hormones, making receptors for estrogen, making receptors for a whole bunch of different hormones, showing the epigenetic changes there. That's how you go from mom's differing maternal style to lifelong differences in expression of all sorts of genes. How's this? What you wind up seeing there as this permanent mechanism, it is also reversible, what he has since shown, which is you have a baby rat who spends the first half of its infancy with some totally terrible, negligent, distracted mom who pays no attention, doesn't do any. Now, cross-foster the pup to a more attentive mother, and you can change the epigenetic pattern. So all of this has two themes going on: early experience causing really persistent differences in how this stuff works long after, and experience later on having the potential to reverse some of this. All of this stuff, once again, would be mistaken for genetic. What we have here is what appears to be a genetic style of what sort of mother rat you are, and it's not genes, it's the mothering style setting up the offspring for being a similar type of mother. Incredibly important studies demonstrating this. What remains unclear is how you get from mom licking you to something epigenetic happening here. His clue is primarily okay, so what do we got at this point? We have gone through over and over here. What have we gotten through at this point? We've gone over and over here at all of the classical techniques in the field of behavior genetics. Does it run in families and adoptive studies, versus monogenetic twins, twin separated birth? We saw all of the problems with it, most dramatically, most excitingly these days, prenatal environmental effects. We saw that trying to separate maternal prenatal effects from paternal genetic effects hits a wall when you get all of these weirdo hereditary things, including potentially non-genetic Lamarckian inheritance of a trait. And what we've seen is how this stuff playing out early in life has multi-generational consequences. What we're going to pick up with on Wednesday is now looking at how people in this business find the actual genes and ultimately gene-environment interactions that make the last 2 hours basically irrelevant. Okay.
For more, please visit us at stanford.edu. Stanford University.
Let's get started.
Stanford University.
Something incredibly cool I just found out about yesterday, which, oh, I wish I had known about like 2 days ago, but still, it's useful. You remember those Siberian foxes and that amazing thing, you breed them for tameness purely on a behavioral trait and come back like 30 generations later and they look like puppies. They have the short muzzles and the big roundy eyes and the cute ears and they wag their tails and all of that. Punchline there being, number one, evolution can move really fast. Number two, in some interesting, mysterious way, if you are selecting for certain behavioral traits, in this case, one where you like being around humans and are all cuddly with them, what you're also going to select for are a whole bunch of traits that are associated with baby wolves in terms of physical appearance. Turns out there's a flip side of this going on right now with what are known as metro dogs, which are dogs that live in the subway system in Moscow. And I don't know, but they live in there, and a whole deal is made out of the number of them which get on subways and know which stops they're going to, and there's an entire website devoted to metro dogs, which are in Russian, but it has lots of nice pictures. But what they're seeing there is that these are dogs who essentially have been feral and in feral packs for decades and decades, and what have they been being selected for? Being able to scrounge, being in fact quite scared of humans, a lot of the time staying far from them whenever possible because they are raiding garbage cans, who knows what. And what they're seeing in the metro dogs is, about 30 generations into it now, their tails are looking more like the tails you see on wolves. Their muzzles are getting longer. Their coat patterns are beginning to get less fancy and distinctive from all the different strains of dogs. These guys are being selected for the exact same traits that went into being wolves. So really interesting demonstration. Again, these guys are not surviving because they've got a different shaped tail. It's just part of the package. If you were going to have this canine-like thing and be selected for being scared of humans, functioning in packs, having a fairly aggressive temperament, you're going to wind up looking like a wolf after a while. Same deal again, very rapidly. So unexpectedly, Russia turns out to be the motherland of all sorts of interesting things with puppies and puppies in the past and puppies in the future and all of that. So that's wonderful. Okay, however, I don't have any pictures of them that I brought today, but go to www.metrodog.edu and see what they have to say. Okay, so picking up on Monday's lecture, I recognize Monday's lecture was probably the first one officially to be able to blow half of you guys out of the water because it was really tough material. Not only was it, as promised, yet another disciplinary jump, but it was a first wave at a lot of material that went by fairly fast and a lot of it quite subtle. What are the things to focus on in there? What did we cover Monday that's critical? Behavior genetics as an overall approach is not doing the constructing an evolutionary story, who's got the best story, we win. It's not molecular genetics looking at evolutionary change driven by actual mutations down at the molecular level. Instead, it's looking for patterns of behavior that go along with relatedness. What we saw was the most completely useless version was, well, here's a trait that runs in a family. And it runs in a family more, the more related the individuals are. Completely useless, bringing in the first of the problems that we're going to have nonstop in the subject, ruling out different environments. We saw a number of the classic approaches they have, which is studying monozygotic versus dizygotic twins, studying adopted individuals, or studying the gold standard of identical twins separated at birth, adopted, and then brought back together again. And in each one of those cases, that's explaining some degree of the confusion, eliminating some of the environment, but over and over and over again, this problem of environment are sneaking in all sorts of interesting ways. Adoption is non-random in terms of where placement occurs, all sorts of stuff like that, making it harder to tell. What we then transitioned to was dealing with the most interesting realm of environment having influences, which is this whole world of prenatal environment. and environmental effects there. One version of it being this whole new field, fetal origins of adult disease, the fact that early events, fetal environmental events, can have lifelong consequences, even multi-generational as we saw, and people are understanding the nuts and bolts of how that might work, that epigenetic stuff. So we saw that as one a very subtle realm of environment slipping in. And we also saw examples where environment could be occurring within an hour of birth, and where environment is occurring despite seemingly like similar things like the number of math classes you take, environment being very critical. What we also saw, just to make life a whole lot messier and more complicated, was violating the rule we have all known since infancy, which is you get equal genetic input from each parent. The business about mitochondria coming from your mother. The business about, as we saw, a more subtle version of it, the same equal input of genes perhaps, but the regulatory control is much more coming from the mother by way of the transcription factors, all of that, and the cytosol of the egg, sperm having nothing interesting going on from a cell biology level, the egg having all of this, and where you can even get something as nutty as inheritance of an acquired trait, Lamarckian evolution. Okay, so that was plowing through all of that, and I'm fairly certain that reviewing the last 2 hours and these last 3 minutes has not put anybody back in the water who was blown out of the water, but so go to sections, this is difficult material. And it's going to get worse today. Okay, this is hard material, and make sure you read the extended notes, because there's a fighting chance that will be more coherent in the lecture. Okay, so we got to the point last time around of seeing that, okay, prenatal effects, the last thing we focused on was actual nuts and bolts of it, epigenetics, all of that. Now we transition to what became the much more modern, exciting version of behavior genetics, unless you happen to have yourself hundreds of pairs of identical twins separated at birth, which is to go and actually find the gene. Finally beginning to bring together, to marry the classical behavior genetics approaches with molecular biology. And we're starting around the 80s or so that people began to be able to work in actual molecular techniques into this whole field. And it started off in a very primitive way and has gotten much better since then. The version that you can start off with is you know something about the trait, or, as we're really saying here, you know something about that there are differences in the trait among different individuals, and now you go looking for the gene. How do you do that? The first classical way of doing that is that you actually have an observable difference in two individuals and two populations and two branches of the same family, a difference in an external manifestation of what's going on with genes, a difference in phenotype, appearance, behavior, some such thing. And what you then do is you use that approach to try to then find genetic differences. You get a whole bunch of examples of folks with one version, a whole bunch of folks with another version, and you start looking for where the differences are. Slightly more constrained version, you don't actually have the phenotype. You are not looking at eye color. You are not looking at how wrinkly somebody's face is. You are not looking at behavioral traits. You're not looking at anything external. The next sort of more focused step is now you're looking at proteins, what some proteins do. Here's some enzyme that turns this into that, and there's two different ways the enzyme could do it, or two different variants on it. And now we got this bunch of people where we take their blood and look, and they've got this variant. These guys have this version of how the protein goes about its business. Can we find the genetic difference? Next step down, next step more frustrating, when you can't see external differences that you're tracking. There's no phenotypic difference that you've got to work with, where you don't know what the protein does, what its function is. Next, lower rent version is to look at the same protein that comes in slightly different sizes, because that's usually a hint that there are different functions. The way the protein works is going to differ, and somewhere along the way there you came up with one version by having some insertion, mutation, or deletion or some such thing. You don't know what the protein does, and you certainly don't know what that does in terms of the individual's appearance, behavior, phenotype, but at least you got something to work with there. So what it is you're using as your starting point, easiest external signs. Next is what would be called intermediate physiological endpoints. Intermediate, it's something functional, but it's not functional like how the person functions. It's functional like how an individual protein functions. And then if you're up the creek with that, at least hopefully going for differences in the size of the protein, the electrical charge of the protein. For non-chemistry types, how this version of the protein interacts with a water environment versus how that version, you don't know what the protein does, but at least you got something to work with, you got a difference. In all of these cases, the strategy is to then see what are the gene differences that go along with it. Primitive initial version would be as follows. You've got, for example, in this case you were trying to find the gene for some disease. And this is where the whole field started, because it is a lot easier to go after a disease. Either you have it or you don't if you pick the right kind of awful disease, rather than going on a phenotype like television taste. That one's going to be a lot harder to find the gene for. The whole field started instead looking at extremes of really major league, you got it or you don't have it diseases. So you've got this family that has this disease running in the family. PKU, phenylketonuria, for example, or Huntington's disease or cystic fibrosis. These were some of the first ones to go. And what you now do is you got the big family, and what you want ideally is a big, huge family where about half the people have the disease and half don't. And the initial way the whole approach would be done was you get blood samples from everybody there, and you start sifting through everybody's DNA, and you're looking for a stretch of DNA that everybody with the disease has in common, and that none of the relatives without the disease have. And what you've got at that point is what would be called a genetic marker. Because doing it this way, you're just sifting through a haystack. You're just trying to pick up big, crude patterns. The difference that you wind up seeing between these two populations of relatives, almost certainly, in fact, as far as I know, never once with this approach, had you just found the individual gene, you found a stretch of DNA that's got 10, 20, 30 genes in there, but somewhere in that stretch is the interesting one. So at least you have a genetic marker here. You know the genetic neighborhood where the difference occurs. So this was, at one point, cutting edge, a technique called RFLPs, restriction fragment length polymorphisms. Do not write that down. And this technique was incredibly slick at some point early on, and it is not easily done because you are dumping 20,000 puzzle pieces over here and 20,000 there, and you're looking for the one difference between the two that's otherwise identical. It's that sort of task. Nonetheless, using that approach, people began to get disease markers, where they got the stretch of DNA that contained a whole bunch of genes and who knows what else, but at least the candidate gene, the gene that was floating around that was responsible for this disease, was somewhere in the stretch of DNA. At that point, what would be done is a number of things. First is people would obsessively check their statistics to see just how certain they are that this is the genetic marker for this disease, because you are soon going to be advising people whether to terminate pregnancies based on genetic tests like those. This is an area where more stringent rules were imposed before you announce you think you know how the universe works with respect to this disease. Really, really major room for things going wrong in terms of it being then applied clinically. So first big demand would be you really have to be much more certain, a much more demanding field than lots of others in biology. The next issue would be this bioethics problem, which is you got somebody from one of these families who may or may not have the disease. This is a disease that doesn't get you until age whatever, and you're younger than that, and this immediately exploded this whole issue for the first time. Should people get tested if they have the genetic marker for that disease, if they are a candidate for it? And this opened up a whole world of confusion about this, where a key feature of these debates was the fact that you haven't gotten the gene, you've just gotten a candidate stretch of DNA, and there remains a very small statistical chance that you've got the wrong one. For a bunch of reasons, there's that possibility. So do you let somebody take a test? Can people tell the difference between a yes and no and a statistical probability? What do you do if it's a disease where the person has just spent their last 10 years watching their parents slowly die from it, and there's no cure for it, and it's absolutely horrible? Do you give them a test that is, if the bad outcome is going to throw them off the top of a building with a high likelihood? Enormously complex bioethics issue. The third challenge, though, was to step up the science from just having the stretch of DNA with a bunch of genes in there to actually find the gene. And in the years since, the molecular biology has gotten much, much better at moving past finding a genetic marker. We are 99.9% certain that the gene that's relevant is somewhere in the stretch, to finding the actual gene and then being able to see what's the difference in it between them and them. What's the difference down to base pairs? Where is the mutation? Where's the difference coming from? That's the world of sequencing genomes now. That's where the field is at. So that has been enormously powerful in terms of potentially finding one single gene. But everything that came through in the molecular lectures last week should be pointing out there's not a whole lot going on in there that revolves around one single gene at a time. So a whole bunch of really horrible diseases, yeah, one gene, single mutation. But most of what goes wrong, if you want to start understanding the genetics of psychiatric disorders, of diabetes, of late onset dementias, things of that sort, it's not going to be just one gene. So the next huge advance in the field was being able to do searches like these for multiple genes. One thing that has been used is a technique called microarrays, gene microarrays, and they were basically invented here on campus by a guy over in the med school named Pat Brown, who is already very famous for having done this, and I suspect has all sorts of interesting awards coming for him down the line. This spectacular technique, you take what is now called a gene chip, and through techniques that are absolutely boggling and make me feel queasy, what you somehow manage to do is you get all the copies of RNA that could be made in a cell from this individual. If you are completely new to this and you've managed to get in this far without having to think about what RNA is, ignore it. This is just sort of a subtle detail for people who are interested in this. What you do is you get a copy of each type of RNA made in a cell. You force the cell basically to transcribe all of the genes that it has, and then you get this little chip, and you basically glue the end of each one of these RNAs to it, and you will have a chip with 20,000 of these little things, and then you use probes which will allow you to see which versions of 20,000 of those genes are different in this individual from that individual. Totally boggling, has forced an entire new world of trying to analyze data, this new field of bioinformatics, because you're not looking at one gene at a time, you're looking at patterns. And that becomes a lot harder. That becomes incredibly important because, again, it's all sorts of interesting stuff where it's not going to be one gene, it's a dozen, it's 100. So that has been one challenging approach. Another is a technique called QTL's quantitative trait loci. What that basically is built around, that if you have subtle enough of tests, you could figure out this is a complex trait that is modulated by 2 genes, or by 3, or by 17. You can see all of this going in the direction from techniques that would let you know you're kind of in the neighborhood of a gene, to actually identifying a gene that differs between the two populations, to a much more useful, realistic world of whole bunches of genes, networks going on at a time. So using these approaches, people have made lots of progress. Sort of the most recent version of that, I mentioned the other day, one type of mutation, a macromutation, is this business about variants of major stretches of DNA, copy length variants where a whole stretch of DNA is duplicated twice or 11 different times. Can you identify traits that are occurring because of differences in the number of copies? Yet another version of dealing with this whole hierarchical, multi-level business about gene regulation. Okay, take-home message from this is 20, 30 years ago when all of this started, you can start with a trait, and through increasingly fancier techniques, you could find the neighborhood of where the gene is, that the different versions of the gene correspond to the different versions of the trait, moving from patterns of that to the neighborhood of the gene, to the actual gene, to whole bunches of genes, to macro differences there. That's one approach. The other approach to the field has been starting the other way. You got a gene. You've got a gene, and thanks to researching it in flies and worms and rats and all of that, you kind of have a sense of what the gene does. And you have a reasonable guess of what sort of traits might be relevant in a human, and now you run it the other way. You have two different versions of the gene, and you look for differences in behavior, or differences in protein function, or differences in the size of the protein. You start with the genetic knowledge. And for our purposes, where people are mostly looking is at behavioral endpoints. So what do you see with that? Lots of findings in recent years, and my feeling is this is one of the more interesting ways of doing this behavior genetics, starting with basic research with animals. You got some sense of what the gene does. How about it in people, because there's variability? First example, and this has to do with a really interesting hormone that we will hear plenty about down the line, a hormone called vasopressin. And vasopressin in rodents, I think I already discussed, vasopressin in rodents has a receptor, the vasopressin receptor. And what we talked about the other week is, depending on which version of this you have, if you happen to be a vole male, it will determine whether or not you are monogamous or polygamous. That's interesting. What was also interesting was that difference, the two variants were not in the gene itself, but in the promoter. And that winds up meaning that the promoter caused in one strain the gene to be expressed in different parts of the brain than in the other version of it. Okay, so what we saw there was all sorts of hints from the animal world that vasopressin has stuff to do with social affiliation, things of that sort. So what do you now do if you want to apply this knowledge to humans? The first thing you need to ask is, well, do we know where the gene is in humans? And with modern sequencing of the human genome, you've got it. So you know where the gene is for the vasopressin receptor. And the next thing you have to ask is, does it come in two different flavors? Does it come in two different flavors, and do they differ in the same way as the ones that people have studied endlessly in rodents? Yay, yes indeed. The receptor gene is exactly the same in everyone, but there's two different versions of the promoter, the same exact story. So that's great. If you've gotten that far, now you start looking for what the differences are between people who have one variant versus the other. And you got to do your homework. You got to match them for age and gender and number of cavities and all sorts of stuff like that. And when you've got that under control, you go looking for behaviors. What's the limit of this? By definition, you're only looking where the light is shining already. You're only looking where you already have a sense that maybe you should be looking, unless you're totally crazy obsessive and want to try to measure everything possible in people and look for a difference. In most of these studies, you kind of know where to look. That immediately means there's all sorts of areas you're not looking. Nonetheless, with this approach, people have gone looking in humans at these different variants on the vasopressin receptor gene, and you find some really interesting differences. One, was a paper published a couple of years ago in a very prestigious journal, which got picked up in the papers all over the universe, showing if you have the version of the promoter that you find in monogamous male voles, you as a human male are likely to have more stable relationships than if you have the other version of it. Whoa, that's pretty bizarre. That's pretty cool. Next thing, a study that just came out in that same prestigious journal a couple of weeks ago, and this is one we're showing which, depending on which version you have, how good are you reading facial expressions in other people, subtle differences in facial expressions. Then there's a whole other world of approaching it, evidence emerging that in families where autism runs majorly through the families, people are seeing mutations in genes related to vasopressin and the vasopressin receptor. So this is one really cool example of starting with that approach, and this is a gene whose variants have something to do with some pretty meaningful realms of human social connectiveness, variability stuff. Totally interesting. Another example. This is a gene coding for a protein called BDNF, brain-derived neurotrophic factor. Doesn't really matter. The main point of BDNF is it prompts neurons into growing new processes. And it turns out BDNF does something that ultimately is kind of a drag for you. In one part of the brain, the amygdala, which I think I've already mentioned, the amygdala has a lot to do with fear and anxiety and that sort of thing, and we will come back to that, don't panic. But the main thing is BDNF plays a role in causing individual rats, when they are highly stressed, to make the neurons in the amygdala grow new connections, new processes. What are you doing? You are training that rat to be anxious. You are training that rat to be hyper-responsive to scary things. BDNF appears to play this role. So now the next step in the sequence, and you'll begin to see it's the same pattern over and over, people then found, there's a couple of different flavors of BDNF. There are a couple of different genetic variants. There's a couple of different ways in which BDNF pops up in rats or mice or whatever, differing by a base pair here, an amino acid there, where you then do your scut work and show, okay, this BDNF works faster than that version of it, or it works less but it lasts longer, or who knows what. You find a functional difference. There's a genetic difference, there's two different versions of it, and it actually makes a difference for how the protein functions, and this has all been studied in animals, and now you understand what it's doing in the amygdala of a rat, depending on which version you have, and then the exact same strategy. Go look at the BDNF gene in humans and ask, well, do you see the same difference? Are the same two flavors in humans popping up as in rodents? Yes, indeed. And then the exact same approach again, looking for differences, and you know where to start looking. What has that literature been showing? The type of BDNF variant that you have, in the exact same way that it maps on rodents, maps onto your likelihood of an anxiety disorder, the levels of stress hormones in your bloodstream, the levels of metabolic activity in your amygdala, same exact sort of finding. Next, a whole other world, which we will learn much more about down the line. This is obviously a quick survey, a whole bunch of different areas. All of these we will come back to big time later on in the course. Next area, this neurotransmitter dopamine. You will learn amazing amounts about dopamine. Dopamine, which works through you guessed it, dopamine receptor. And in fact, there's a gazillion different types of dopamine receptors. And dopamine receptor number 4, D4, the dopamine receptor 4 gene, has something to do with a whole bunch of interesting traits in rodents, and it comes with variability in the rodent version, different flavors, and humans have variability along similar lines. And dopamine, what is dopamine involved in? Pleasure and anticipation and behavior, reward-seeking behavior. What do different versions of the dopamine type 4, the D4 receptor gene, begin to map onto in humans? Levels of risk-taking behavior, levels of sensation-seeking, levels of novelty craving. That's interesting. Another example, a gene called NPY, a protein, a neurotransmitter called neuropeptide Y. Do not get overwhelmed by all of these. It's just the same pattern over and over. NPY, same deal again, and two different human variants have something to do with differences in levels, again, of anxiety, of metabolic rates in the amygdala, all of that. And in this case, the NPY difference is quite interesting because it's one of those where the difference is not in the gene structure sequence, it's once again a difference in the promoter. Okay, so whatever we have in all of these, this is a really powerful approach. The first version that we heard, you've got a behavioral difference, you've got a phenotypic difference, you've got a difference in how two proteins function or their size. Go looking for what's the genetic difference between those who have this trait and those who don't. This version, reverse engineering it, you start off with a gene that comes in different flavors, you've got a sense of what it does from animal studies, do you have the same variation in humans, and does it match up to behavior? So all of these are amazing. All of these, it has to be emphasized in a way that will come back to haunt us also big time. All of these explain tiny percentages of the variability in the data. There's nothing remotely resembling a world in which if you have flavor vanilla of the D4 receptor versus if you have flavored chocolate, if you have flavored vanilla, you are going to be hand gliding when you were in preschool. You're going to be totally sensation seeking, and if you have the other version, you're going to spend your entire life collecting stamps or who knows what. It's not that deterministic. No surprise with this gene stuff. No single gene is going to be particularly deterministic once you get outside the world of phenylketonuria and cystic fibrosis and such. They contribute some explanatory power. So on one hand, amazingly cool seeing these genetic links to aspects of human behavior. On the other hand, they're not very big effects. And this is something we will come back to endlessly down the line. Okay, so this is how the field has gone about more recently, trying to find links between variation in genes, or their promoters, and variations in aspect of behavior, running at both legs. And what all of this is looking at, what is intrinsic in everything we've been talking about with this behavior genetics, is, oh my God, the cliche finally emerges, nature and nurture and gene and environment and gene-environment interactions. What is becoming increasingly clear in the field is there's a third leg to all of this. There's a third leg that could be extremely powerful and is going to be the basis for the chaos and complexity lectures down the line, which is chance, the role of random events, the role of chance in being a form that transcends what we would call environment and is certainly not genetics, a whole different world where that contributes. Okay, where will we see chance playing out an important role in all of this? Brownian motion. Brownian motion, for those who don't have a techie background, I will describe it in the way that I understand it, which is flailing desperately. Brownian motion has to do with the fact that molecules oscillate and there's an intrinsic movement oscillation of molecules. You take a whatever full of water filled with all sorts of things, molecules, whatever, and they're all going to be vibrating to a certain extent because of this Brownian motion. I suspect that was the most amazingly incorrect description of what Brownian motion is, but that's the point. That's the main point, simply that there's this oscillation which is completely random. It is just an intrinsic feature of how the world of physics and Albert Einstein's mustache works, and so you've got this Brownian motion stuff going on. So where might that play out in terms of chance? So we got here a cell, and with our four powerhouses of the cell here, and on the right, let's assume this is a genetically identical cell, and I just got tired of drawing it completely, but the same four mitochondria, and what we see here, first off, is genetically, these mitochondria differ. One of them has a blue spot, one of them has a red spot. Wait a second. Every single cell in your body was built out of the same genome when you started off life. aha, back to that thing the other day, mitochondria have their own DNA. Mitochondria are going about their own business of dividing in the cell, of replicating themselves now and then with a mutation. Within any given cell, the mitochondrial sequence of DNA is going to differ from one mitochondria to another, because they've been like separate organisms that are inside a cell going about their own evolution, their own mutations, their own whatever. What do you then have? What would be called a mosaic of different genetic profiles of the mitochondria within a cell. So that in and of itself is pretty interesting, because this is a whole other world. This is not the DNA back in the nucleus, and this is not any sort of variability introduced by jumping genes or accessibility of transcription factors to genes, all that. This is, meanwhile, outside the nucleus, there's this whole weird world of these mitochondria that are functioning all on their own, so the different mitochondria in the same cell will have different genetic makeups. So great. So these two cells are absolutely identical. Each of them have 4 mitochondria, and each of them have 2 red kinds, 1 blue kind, and 1 that gets nothing. So now that cell is going to go about splitting in 2. And what happens is it splits, and in the process of splitting, what the fluid, the cytosol, the stuff inside, where all the molecules, including the mitochondria and the other organelles, are found, and it splits, and there winds up being a very simple question: which mitochondria wind up in which daughter cell? And what we've got here is, in this case, a split like that. In this case, a split like that. Oh my God, why did that happen? Is that because there is a gene in this one that says when you form a new pair of cells, have blue and blank in one and 2 reds in the other, and this one has a completely different instruction manual for how you split cells? No. Simple randomness of where the mitochondria were in the cell when they split. A totally random event. So what you see here is you start off with 2 cells that are genetically identical, and within 1 round of cell splitting, these genetically identical individuals are already genetically different. How come? Because of instructions or a blueprint? No, simply because molecules, organelles, things like mitochondria just oscillating around, and along comes a split, and who is stuck on which side of the divide, so you've got variability that way. Intrinsic in that is another type of variability, which the second I mention it, you will be able to run with effortlessly, which is floating around. And here also will be transcription factors and splicing enzymes and other enzymes, and along comes the split, and there's not the remotest chance that half of every single type of transcription factor is going to go each way, and half of every type of splicing enzyme, it's going to be who's oscillating where and where with their cell splits. So you wind up not only with different outcomes in terms of mitochondria and their genetic makeup, you're going to have different distributions of different types of transcription factors, all due to this random oscillatory stuff. So that's one big component where chance plays a role. And what the chaos complexity lectures are going to be very heavily about is just how important stuff like this is, how important it is in explaining what goes on, how important it is in explaining why, no matter how much you know about what every single molecule is having for lunch next Tuesday, there's still going to be a huge degree of unpredictability. All of that is to come. This is just beginning to establish the idea, yes, nature, nurture, nature, nurture. Chance also is the third leg, and they're playing a large role. Whether chance is what's going on when you get a transposable event, you remember at the point that neural stem cells are beginning to divide into neurons, that's when they juggle around their transposable genes most dramatically. Whether that is a completely random event as to where the copies land when they come back to Earth, I'm not sure if people know, but I suspect there's an element of chance in there also. So this is introducing that notion of it. So great, so now you've got all these caveats, which is environment is subtle and coming all over the place, and early experience and epigenetic changes, and all the different ways you could find genes linked to behavior. And don't forget, variability also has something to do with chance. At the end of the day, though, what all of the behavior genetic approach is going to come down to at the end is a single number. We have studied this trait with adoption techniques, with looking for genetic markers, with looking for copy length variants. We have studied this trait and the variability of it, and thanks to us studying for a gazillion years, we can now conclude that 53.5% of the variability in this trait is heritable. You come up with this number at the end. This trait has a 53% heritability. That's the number that winds up coming out. Scientists report that this or that trait has variability, has 80% heritability. This is the number that always comes out at the end. What's the heritability? Ranging somewhere from 0% up to 100%, this is heritability. And this is absolutely critical, because this is a truly influential number, the one that people come away with when they are being taught, oh, this trait is genetic. Those who want to be quantitative will have been told, whoa, this trait has 80% heritability, it has 90% heritability, all of that, and it's the wrong interpretation. And what we're going to spend a bunch of time on now is looking at what heritability actually means. The heritability number, because it is completely different from the everyday intuition about what it means, and what it's usually telling you is how unimportant genes are in a deterministic way rather than the other way around. Okay, so what does everyone assume? What is heritability telling you? It is telling you how much do genes determine the average level of this trait. You have one version of a gene, and there's the other flavor, and they produce different average levels of some behavior or something at the end. What heritability is teaching you, if you've got this completely wrong notion, it's teaching you how much do genes have to do with the average level of that behavior, that trait, that whatever. That's not what heritability actually means. What heritability as a number is telling you is not what genes have to do with the average level of the trait, it's what genes have to do with the degree of variability around that average. Okay, this is a point where things immediately seem panicky and start getting very, very complicated, all of that. We will see it's not that bad. And it's not that bad in a very critical way. Okay, so simple first sense of getting at it. Here's two populations where you measure something or other, and these three individuals, they come in at 9, 10, 11, and these come in at 1, 10, and 19 or whatever. What's going on here? You average them, they have the exact same average. What's the difference? There's a lot more variability around the average here than in this case. What's the wrong idea that people have that heritability means? Ooh, it's telling you how much do genes determine the average here. That's not what it's doing. It's how much do genes determine how much variability there is. Initially, this seems like a very subtle point, and what's the big deal? Because at the end of the day, it's still talking about how important genes are. What we'll see now is it's actually a way of seeing how less deterministic genes are in lots of cases. Okay, so this heritability stuff talking about variance. Here we have an example. Okay, we've got some plant with some gene that comes in 3 different flavors, and you measure something or other about the plant. This is how much water it retains, or the plant's IQ, or something like that. You're measuring some trait, and you're asking, does it differ as a function of which version of this gene you have? So you do your study, and this is what you see. And you say, whoa, okay, I went and looked at this plant in a rainforest, and we identified the genetic versions of it, and look, very different. The gene that you have there, knowing that, gives you a lot of predictive power over what level of whatever it is that you're measuring this plant is. That's great. So you're going to get your doctorate out of that, and you get some publications, and it's great, and you finally stop being a student, and it's terrific. And meanwhile, some individual who shares like none of your genes in common on the other side of the planet, meanwhile, there's some SOB who's studying the exact same plant in the middle of the Gobi Desert. You're sitting here in the Amazon studying this, and here's this individual doing the exact same kind of experiment, saying, oh, here we have this unlikely plant that grows both in the Amazon and the Gobi Desert, but here we have this, and we're studying this here, and what am I studying? I'm studying those three variants of this gene, and I'm asking, do they influence plant IQ? The same thing that you were asking over here. And that individual is doing their study, and they see that yes, indeed, the gene influences what version that you have. And here you got A, B, and C, and here's what they see when they measure plant IQ. And it looks like that. And what do they conclude? Whoa, look at that. Knowing what version of the gene you have gives you enormous predictive power in predicting what the plant IQ is going to be. And then, catastrophically, tragically, the two of you meet each other and discover that you're both researching the same plant, and you look at your numbers, and your heart breaks at that point, because you look at these, and what's going on here? What's going on? Let's translate this notion of heritability and variance and all of that. Let's translate it into a very simple question you can ask, translating all of this in English. You're interested in what this gene has to do with plant IQ. And you're allowed to find out one piece of information. You could know whether the plant has version A, B, or C, or you could know whether the plant is growing in the Amazon or the Gobi Desert. Which piece of information is going to give you more predictive power? And what you wind up seeing here is, if you can either know this or the environment, you want to know what the environment is. The variability, plant IQ could come in at 98, 100, 102, 8, 10, and 12. Far more of the variability in those 6 numbers is explained by what environment it's going on in rather than the genetic difference. That's what heritability is telling you, and in a study like this, it would tell you that the heritability number is actually quite low, because the amount of variation due to this is far less explained by the gene type than whether it's the Amazon or the Gobi Desert. Okay, so that's a first pass at it. Why is this really important? For the following reason. You're a scientist, and you're trying to understand how A, B, and C influence plant IQ, and you come up with something nutty and stupid like saying, well, I would like to be able to do field seasons in both the Amazon and the Gobi Desert, and your advisor will say, or all of your elders will say something like, no, you can't do that because that's just, you're not controlling for environment. You're not studying it in only one controlled environment. That's too, you can't go to, there's such different ecosystems, all of that. Pick one and go study it there. And what you do is you go study it there, and as a result of just studying it here, you come away thinking that virtually all of the variability is explained by genes. What have you just done? You explicitly have designed your experiment so that you can't detect the environmental role in determining that trait. What counts as setting up the experiment as the right way for people who think about this sort of thing, standard sort of approaches to experiments, what counts as doing a good job by definition, what you're doing is biasing towards thinking the genetic input is more important than it actually is. Because scientists don't decide to study this trait in 2 different places. You don't decide to do a study where you're looking at some trait in both rats and ocelots or something. You don't decide, whatever. Look, pick one species, maybe even pick just one gender, pick one environment. Don't have your rats in one room where the air conditioning is going nonstop, in the other room you're barbecuing stuff. Control for environment. And by definition, if you've done the nice, careful, responsible thing that a scientist is supposed to do, doing this sort of thing, you control for environment. What have you just done? You have just removed your ability to see the role of environment. And you've just artificially inflated how important you think the genes are. Okay, let's take a 5-minute break. We are going to look at this in a lot more detail. Do not freak out if this is not immediately intuitively obvious. Questions that just came up. The first one is pointing out one of those where I probably should have actually taught something clear. What are you talking about with proteins being different sizes? How do you figure out proteins are different sizes? And the answer is you make them stand against the wall in their bedroom with a ruler and you mark it there, and then you see if they differ in size. Or if that doesn't work, what you do is you use various biochemical techniques. You basically make something akin to a thick, homogeneous soup of something or other, and you put the two different versions of the protein in there, and essentially you sit and wait and see how far they sink down. And the one that's heavier is going to sink down further. Anyone who knows about gels and electrophoresis, that's insanely simplified, but that's basically the notion of it. Put them where they move through something as a function of their weight, gravity, and you could then pick up size differences. The other good question that was asked is, what did you just say in the last 10 minutes? This is very difficult. This is an extremely subtle point, and I'm going to hammer it in over and over again here in sections, all of that. Why is it so important? Because number one, this is a number heritability that the general lay public comes away with interpreting completely wrongly. And number two, the vast majority of scientists, when they're working in this field of behavior genetics, design their experiments so that by definition, they are eliminating all sorts of realms of environmental influences. Okay, so going back to this here. So here we see once again this simple question. Use this one over and over, translating all this theory and variation and stuff, always translate into the same question. I can find out what version of this gene, or I can find out which environment this is happening. Which one do I want to know if I want to be in a better position to guess what's going on? And when it looks like this, you don't want to know the genes, you want to know the environment, because it's far more powerful. And if you only studied this in this setting, you would come away saying, oh, this variability is entirely explained by this version of the gene. Oh, this trait, plant IQ, has 100% heritability. And what you see there is when you combine it together with numbers like these, it's 15% heritability. So that's totally critical to hammer in. Okay, so what are some of the responses at that point by people who will say that's ridiculous if you're saying what heritability mostly should be teaching us is how unpowerful genes are? What would be one of the initial responses? Great, how many plants out there are growing in both the Amazon and the Gobi Desert, and it's so hard to study IQ in plants? This is a totally artificial dichotomy between the extremes of environment. You're like inflating things now, you're cheating in the other direction to get the most dramatic artificial circumstances to inflate your sense of how important environment is. This is totally artificially dichotomized. So think about humans and think about one single fact, which is we inhabit more different environments than any other species on Earth. We live in the Amazon, and we live in the Gobi Desert, and we live in Peoria, and we live in all these things, and we have more exposure to different sorts of environments. So immediately that argument goes down the drain. Okay, so now instead somebody argues something different, saying, okay, yeah, sometimes plants have IQ in the Amazon and in the Gobi Desert. And I get your point, your stupid point here that, ooh, environment can make. No one's going to argue that the difference between the Amazon and the Gobi Desert isn't important. Oh yeah, okay, well humans, they live in both, all of that, it's not an artificial difference. But you notice something? Isn't it interesting that in these two different environments, C plants always have the highest IQ and A plants always have the lowest? That's telling us something about that gene. That's telling us, and you're then saying, well, yes, it differs by environment, but we've just learned something very important about these gene versions, which is in totally different environments, version C gives you a higher plant IQ. So that's important, yeah, environment, but we've just seen how powerful this gene is. But then you run into the person at the conference who is studying it in the Gobi Desert, and they put their data up, and it's even worse than in the last version because it looks like this. And what have you just learned? You can't say a thing about this gene. You have just learned the translation of this sentence. The first critical sentence we've had over and over is, if you can only know one factoid, one about which gene version, one environment, choose the environment. We have now just learned a second sentence, a second question to ask, which is, what does having A, B, or C have to do with plant IQ? And if the answer is, it depends, you've just seen the subtlety. If the answer is, it depends on which environment you're looking at. If you're looking in the Amazon, C gets you the best plant IQ. If you're looking in the Gobi Desert, C gets you the worst plant IQ. What have you just shown? What is technically the definition of a gene-environment interaction? And we've just seen going from, well, yeah, they're very different, but C is always the best. Isn't that interesting? To a completely different profile. What does A, B, or C have to do with plant IQ? It depends. It depends on the environment. That's how you've just defined, that's your diagnosis for gene-environment interaction. And what ultimately one has to argue is that it is impossible to ever say what a gene does. You can only say what a gene does in the environments which, to date, it has been studied in. Okay, let's see that expanding even more. Because you've got this. Okay, let's jump ahead. Okay, so this is showing you now just how totally nutty and counterintuitive heritability terms actually are. You ask a question, what's the heritability of number of fingers on your hand? You know, genes have to do with the fact that we got 5 fingers instead of flippers or some such thing. Genes have huge amounts to do with it. You're not asking about the average number of fingers, you're asking about the variability. Remember that again. So what are the circumstances out there which will give somebody 6 fingers instead of 5? That's incredibly rare. What about 4 fingers instead of 5? Oh, industrial accidents. 3 fingers instead of 5, lots of industrial accidents. 2 change jobs or whatever. What are we seeing here in terms of how much do genes have to do with having fewer than 5 fingers? It's all industrial accidents. Genes have nothing to do with it. There's no doubt some weirdo disease out there, but for our purposes, this is how it works. What have we just discovered? Number of fingers, that trait has a 0% heritability. That's totally bizarre. That's completely counterintuitive. Genes have everything to do with why the average human has 5 fingers, but they have nothing to do with the variability. In that case, it's entirely environmental. The number of fingers you have has 0% heritability. Now let's look at another example. It's 1950 in Eisenhower, America. Actually, it wasn't until 1952, but it's a very different world than now. And one of the things that you would never, ever, ever, never see in the United States would be some guy wearing an earring. Unless in a very cloistered part of the country he was a sensitive guy, but for most of America, this is not what men do. They don't wear earrings. And likewise, in most of America, if you were a good red-blooded American woman, you would not go outside without your earrings on. So now you gotta say, okay, well, what causes variability in ear ring wearing behavior? And it's entirely explained by whether you are female or male, which is a genetic trait. What we've just seen is whether or not you wear earrings in 1952 has 100% heritability. Totally counterintuitive. Think through this again and again and again, because this makes sense. When heritability is a number about this rather than this, you get a world where 0% heritability for your number of fingers and 100% heritability for whether you were wearing earrings at that time. Because, once again, asking, okay, I've got a choice in the matter. I can either know the entire genome of this individual, or I can know whether they are in a frat where they close their eyes and work with a wood saw every now and then. Which fact do I want to know? That's the one that will tell me what about the environment going on with them. Or now you have a choice. I can either know the entire genome of this person, or I could know that what sort of environment they're living in 1952. What do you want to know there is male or female. If I know that, I can completely predict this behavior of earring wearing. So this totally counterintuitive thing here where heritability is telling just the opposite of what people intuitively think. And as soon as you deal with that and recognize the way scientists do experiments is to try to do things as cleanly as possible, study it only in one place, in only one setting, in only one circumstance, you've just artificially guaranteed that you're going to come away more impressed with the genes than they actually deserve to be. So how would this look? Beginning now in more detail. So what we've got here are a number of different ways in which you can see when are genes important, when are environment important, that sort of thing. Okay, so we have here two different traits. No, we have one trait, two different flavors of the gene, and flavors A and B, and three different environments. So here you have the Amazon, the Gobi Desert, and a roller coaster. And you're measuring plant IQ, and there's two different versions of the gene there. And you're asking, well, what does your environment have to do with it? Your data look like this. What does it tell you? Environment makes no difference at all. It doesn't matter if you're in the desert, the rainforest, whatever, which version of the gene you have entirely explains the variability. So this is what a heritability of like 100% would look like. Now you do the study, and instead you get data like these. You'll note just one of the most important things about being a card-carrying scientist, which is data are plural, which talk about counterintuitive. Earrings are genetic and data are plural. So now you get these data, and you see as follows. In this environment, no difference depending on what type of gene you have. In this environment, no difference, no difference. In each environment, very different averages. This is what it would look like with 0% heritability. There's no difference at all of the variation explained by gene variation. It's all environmental. So now we have a version that forces us to put in the same phrase we heard about before. This is what your data look like now. And you now ask the question, well, what does being in environment, what does your environment have to do with your plant IQ? And the answer is, it depends on which version of the gene you have. And now you ask, what does having a certain version of the gene have to do with your plant IQ? And the answer is, it depends on which environment you're in. This is, in a sense, the verbal definition, again, of a gene-environment interaction. What do genes have to do with this trait? Depends on the environment. What does environment have to do with the trait? Depends on the type of gene. This is what the data would look like. And here you have an interaction, and in some fudging of the numbers, this is what 50% heritability would look like. What we just saw here before, hypothetical example of the messiest it could get, this is the one where the people come back and say, yes, environment matters, but you notice version C is always associated with the highest IQ. And here we have the version where, depending on the setting, if you have this type of gene, gets better. If you have this type of gene, it gets worse. This is one where there's a dramatic interaction between what the genes are doing and what the environment is doing. This is one that's going to have an even lower heritability, because this is one where you are saying big time, it depends. It depends on what the gene type is. It depends on what the environment is. This is, in a sense, as dramatically as it could be that case. So you constantly have stuff like this going on. Let's look at an example where, in fact, you wind up seeing something like this. Back to our iconic mutation disease from the other day there, PKU, phenylketonuria. Just to remind you, do not worry about the details. It's this disease where normally there's this thing in your body which would be toxic to your brain unless it's turned into something else. And this is the enzyme, the protein, and thus coded for by a gene. This is the enzyme that turns the scary version into the safe version. And in PKU, you have a mutation in this enzyme. It's not doing its job. The scary version builds up and wipes out your nervous system. That looks bad. And what we've got now is a trait where you're going to say, well, let's see. I'm very interested in knowing, being able to predict whether or not this person has a fairly normal looking brain or whether it looks like Swiss cheese. Do I want to know whether or not they're growing up in Idaho versus Montana, or do I want to know whether or not they've got a mutation in this enzyme? Obviously, you want to know if there's a mutation in the enzyme. Seemingly, heritability is 100%. But now you do something which people have been doing for people with PKU for a couple of decades now, which is you put little labels on bottles of food and stuff saying if this food contains the scary bad news thing. And if you have PKU, if you simply don't eat food that contains that, if you have what's called phenylalanine-free diets, you don't get your brain looking like Swiss cheese. What have you got there? You've just put in an environmental influence that has reduced heritability down to 0. This is a case where we've got something like this looking. And here we're measuring is like the Swiss cheese index or whatever of the brain. And what we've got here is this is an environment where people know how much of the stuff there is in different types of food and don't eat it. A simple environment, not so simple, but an environmental intervention, a behavioral change in the environment, and you've just reduced heritability from 100% to 0%. So that is a dramatic version. Now, let's look at a version of a real-life example of this. And this would be with, I think I've already put this one up. Okay, does this look familiar by now? No, okay, that was it, Thanksgiving. Okay, what we've got here is a gene that's got something to do with depression. It's a gene that has something to do with the neurotransmitter serotonin. Details don't matter, they will come later, but the gene comes in 2 different flavors. And one version of it, based on everything that's known from laboratory animals, one version should be the kind that predisposes you more towards depression. So in this incredibly important study that was done a number of years ago, a guy named Caspi, Duke University, and colleagues, they had been studying 17,000 people, basically from birth up to age 25 or so in New Zealand, and who knows why in New Zealand, but studying them there and following and asking, they took blood, so they know the whole genetic profile, and looking at, among other things, by age 25, does the person have latent depression or not? Have they had an episode of depression? And now let's go look at which version of this gene they have. And what would everything predict from all the animal studies, which is you have the bad version of the gene, and you're, as a human, going to have a higher chance of having a depression. That's not what the data looked like. Instead, it was one of those, it depends. It depends on the environment. What you saw was it all depended on how many major childhood stressors there were. Loss of a parent, divorce, abuse, some such thing. In the absence of any of those, this was the rate of depression in the people having the good version. and throw in one childhood trauma to them, 3 of them, and the incidence is going up slightly. Now, you've got the bad version of the gene, and it puts you more at risk only in certain types of environments. So here we have a great gene-environment interaction. That's something straight out of this one. You're saying, well, what does this gene have to do with the risk of depression? And the answer is, it depends. It depends on what your childhood environment was like. But now one additional detail showing that this is actually a case of this, one that just gets ignored whenever people talk about this study because it's cool, but it's actually not all that important. Okay, so here's the people with the good version of the gene, but it makes this point though. You got the good version and they're down here, and you know what's going to happen. The bad version is going to be exactly the same, but you look at the data closely, and in the absence, the bad version is actually a little bit protective from depression versus the quote good version. What have you just discovered there? It's one of these. This is a radical gene-environment interaction. Not only is it the case that does this put you more at risk? Well, it depends on the environment. Depending on your environment, it could put you less at risk. This one is not a big deal because there's a tiny difference. There's no reason to pay attention to it other than just sort of pointing it out pedagogically. But this is an amazing example where this can go in the opposite direction. This is a hugely important gene-environment interaction. So what all of this begins to set you up for is we've got this heritability number, and people come away constantly thinking about is telling you about how much the average level of a trait is determined by genes. It's instead telling you how much the variability. What that means, thus by definition, that there's all sorts of nutty, counterintuitive things going on where earrings are totally heritable, a number of things are not. What that tells you is, by definition, the way experiments are set up to make things nice and clean and interpretable, scientists typically remove environmental variability. Scientists typically artificially have boosted up the seeming heritability of a trait. What does that tell you? The more different environments you study a trait in, the lower the heritability is going to be, because you are going to be giving more and more opportunities for things to be different, more opportunities to be able to say, it depends. It depends on which environment. We've just studied it in 99 different environments, and it always looks like this, no difference by environment, and genes always make a huge difference. And then you go out, and before dying of fatigue, you study it in the 100th environment, and it now looks like this sort of thing. What you've just learned is this is a it depends example. The more environments you study something in, the lower heritability is going to be. Translating that into English, the more environments you study a genetically influenced trait, the less interesting and important the genes are going to be. The less interesting and important they're going to be in answering what does this gene have to do with whatever. And what we're seeing over and over again is the only way to answer it, or over and over again, it's going to be it depends on the environment. Ultimately, as I said a little while ago, ultimately it winds up being meaningless to ask what a gene does. Ultimately, the only really truly scientific way you can answer a question like that is, what does this gene do in this particular environment? People make this big deal out of, oh, genes do something, environments do something, and every now and then, hooray, they interact in some exciting way, and teaching the gene-environment interaction cliche, that's what's happening every single time. And that winds up being the basis of that quote from Paul Ehrlich in this department that I put on the handout, which is great, summarizes this entire point. Asking whether genes or environment have more to do with some trait is akin to asking whether height or length have more to do with the volume of a rectangle. They're inseparable. There's no such thing as a gene influence outside the context of an environmental interaction. Okay, let's look more at what this looks like. Here, this being one great example, and this being this iconic one, and this is a finding that is destined to be like the most important in the last quarter century in biological psychiatry. It is the most powerful demonstration in the realm of abnormal human behavior of what your genotype has to do with your behavior, the most amazing, logical, intuitively reasonable demonstration of it depends. It depends on how stressful the childhood environment was. Really great, bringing things back to last week's sort of molecular lectures, people already know who study this, the two different versions of the gene. Certain classes of stress hormones interact with the two versions differently. When stress hormone levels aren't particularly raised, that difference isn't being manifested. The more of a history of stress, and it's obviously more mechanical, more complicated than just how much you're exposed to, but the promoters, the variation here is in the promoters. The promoters interact differently with those glucocorticoid stress hormones. In the absence of the glucocorticoid stress hormones, because in the absence of something exciting, the genetic difference makes no difference. So we've just translated an epidemiological answer. What does this gene have to do with depression? It depends. It depends on your childhood stress history. We've just translated that into last week's molecular biology. What does this genetic variance have to do with whether or not you have depression? It depends. It depends on how much childhood exposure to glucocorticoids you have. So we've just leaped from analyzing this in the context of this field to translating that into last week and translating that into the endocrine lectures that are going to come next week, beginning to show, okay, you're an epidemiologist, the answer is what sort of environment? You're an endocrinologist, the answer is, well, the hormonal environment. Whether you define it, it's the same punchline. It depends. It depends on the environment. So this is the best, most amazing example of this in all of psychiatry. Except these guys, Caspi and colleagues, measured something else, and they got just as bestest of an answer for that, which is they looked at another gene that has variation, one we will hear about down the line, a gene called MAO, monoamine oxidase. All we need to know right now is a gazillion animal studies suggest that one version of the gene predisposes more towards aggression than the other. Whoa, so they got 17,000 people who've just had 25 years worth of opportunity to be aggressive or not, and they got the genes. And now they ask the question, rather than at age 25, has this person ever had a major clinical depression? Now they ask at age 25, has this person ever been in trouble with the law for some sort of antisocial behavior? Antisocial behavior is the new jargon for what used to be called sociopathic behavior. Has this person been in trouble with the law for sociopathic behavior, antisocial? And does it vary depending on which version of the MAO gene you have? And what you get is, not only is the answer, it depends, the graph is basically identical to this one. It's superimposable, the magnitude of the effect. What was this in that case? How, whether or not you had, and how severe your child history was of abuse. The more childhood abuse you had, the more having the wrong version of the gene increased your odds of having antisocial aggressive behavior. Once again, a major dramatic it depends. And it's stunning. You look at these two different papers, both were published in Science a couple of years apart, landmark studies, all of that, and the two graphs really are virtually superimposable, the same magnitude of effect, the same answer. It depends on your childhood. So that is a very strong example. Next one, another one. Now you go back to this gene difference with the serotonin, the depression world, and now you study it in monkeys, and the variation there, there's variation in brain chemistry related to depression, all of that depending on how stressful the monkey's childhood was. Same exact sort of finding. What else? Now you begin to look at, going back to that world of dopamine receptors and thrill seeking and all of that. And what you look at there is variability in a certain type of different dopamine receptor gene. And what you find there is if you have a certain version of that gene, you have less social attachment. There's a whole psychology of how you measure that, and God knows why. It's associated with less social attachment if, and only if, you got brought up by a mother who was cold and withdrawn. There's once again a it depends. By looking at that, it's nowhere near as clean as this, but it's the same sort of theme again. Another example of it, there's another gene called FAD2, and don't even ask me what it stands for, but which version of the gene you have has some predictability over your IQ. That's kind of interesting. It's not a big effect, but nonetheless it is demonstrable. Which version of that gene you have has some control, but there's a it depends. Which version of that gene you have has something to do with IQ if and only if something. Let me tell you what this FAD2 is involved in. It is involved in carbohydrate metabolism. Carbohydrates, carbs, carbohydrate metabolism. It codes for some enzyme that breaks down carbohydrates and no doubt some certain kind. Okay, what's going on here? You've got two different versions of a gene that break down carbohydrates, and you got one version, and on the average you'll have a higher IQ if and only if some environmental thing was happening. Any guesses what that environmental thing might be? High carbohydrate diet, okay, that makes tons of sense. Yes, indeed, that winds up being relevant. Remember, you're now, not remember because I didn't say it already, but you're looking at IQ in kids, so you're looking now at carbohydrate stuff going on early in life, so frame it in that context. So we got a start of an answer here. So what else could be happening? If and only if the version you have of this gene that involved in breaking down carbohydrates, translating into how you're dealing with the amount of carbohydrates in your diet, gets you a different IQ with an if and only if. What would the if and only if be? Build on that comment there. What else could it be? Okay, I'm not even going to look. Somebody shout out an answer. You're right. Okay, who started? Somebody started saying something. Who started saying something? Okay, any other ideas? Who said that? Look at that, they start pointing. The person doesn't even fess up. Who said that? You're right. Okay, you want to be that way? I'm out of here. Okay, so if and only if you read the damn notes, as it turns out. Yes, breastfeeding. Okay, breastfeeding. If and only if you are having a particular type of diet very early in life that is extremely rich in the types of carbohydrates that this enzyme works on. So a very dramatic if and only if there. I don't know why I write that stuff, or I don't know why I show up here. I should be doing one or the other and some sort of environment interaction thing. So yeah, breastfeeding. Okay, so one more example, a very interesting one. And this is one that has at some point or other, I bet, been pertinent to every single person in this room, which is there's another genetic difference that has something to do with a certain cognitive aptitude. And this one, everybody knows about. And this one is as follows. If you have, there's two different versions of this sort of genetic picture, and they're associated with different levels of aptitude in this particular cognitive realm. This is not a world anymore of one single gene at a time, FAD2, any of these. This instead is talking about a whole bunch of genes if you have a Y chromosome or not, if you are male or female, a genetic trait. And what is the thing that has been demonstrated most consistently in the literature forever and ever and ever in terms of a gender difference in a cognitive skill in math performance? That has come through endlessly. We already talked about it the other week. A small difference in the average median performance on junior high school, Johns Hopkins superstar kids taking the SATs, but a big difference in the tail at the end, the high performance, all of that. This comes up in endless studies. This comes up with kids at fairly young ages. There is this gender difference on the average. Yes, we say on the average. Don't forget you say on the average before you even ignore that. You say on the average, boys are better at math than girls. Men are better at math than women. Male plants in the Gobi Desert are better at math than female plants there. And so we got a genetic trait here being a function of, whether or not you get a Y chromosome. But then a few years ago, there was an astonishingly important paper published in Science. And what these people did was look at math performance scores of, I think it was 480,000 different high school kids, and all over the world. They didn't just study it in America. They studied it in 40 different countries. And they asked a very simple question, which is, are there big differences in gender quality of life issues in this country? And there's a whole index that comes out of the World Health, UN, something or other, called a Gender Equality Index, which takes into account, like, if there is, and if there is, how dramatic, of a gender difference in educational opportunities, in freedom of movement, in freedom to serve in an elected office, in freedom to vote, in freedom to choose who you're married to, and obviously the enormous variability on this planet in terms of that. And what they showed was the greater the inadequacies, the greater the difference in gender treatment in a society across 40 different countries, the bigger the difference there was in math scores. It's not a function of gender, it's which society you're growing up with your gender. What was at the most extreme? Let me make sure I get the countries right here. Who had the worst profiles of the 40 countries in terms of the biggest gender differences in these quality of life measures? Turkey, Tunisia, and South Korea. Where was the United States? Sitting somewhere around the middle with most Western European countries. And which were the countries on Earth which, as a bloc, had the lowest degree of different treatment of people in their society based on gender? The ever handy, wonderful, utopian Scandinavians. So in come the Scandinavians, and what you show is, by the time you look at the country on Earth that has the least gender differentiation of any, which is Iceland, you notice something different. There still is a gender difference. In Iceland, girls are better at math than boys. Slightly, a small difference going on there, but nonetheless, as you go from the countries in which from day one, girls transitioning into women are given the most constraints of freedom of life, that's where you're going to see the biggest math difference in scores. And you look at less and less of those sort of inequities, and the gender difference in the math score, by the time it's at the Scandinavian countries, it's down to 0, and then you get to Iceland and it actually reverses. It's got nothing to do with your Y chromosome. We immediately come back to the first of those questions from before, that diagnostic question. You got a choice. You want to have a sense of, in this population, now even at the individual level, you got a choice. You're comparing two individuals, and you want to guess which one is better at math than the other. You can either know their gender, or you could know whether they grew up in Tunisia or Iceland. Which fact do you want to know? You want to know about environment. Environment is vastly more powerful there. Another feature of that has been interesting, so you then say, okay, the extremes. Okay, so in some societies, the means are exactly the same, but what about that difference way out at the highest level of performance? As I mentioned the other day, in the mid-1980s, looking at the highest first percentile of math performance on junior high school kids with the SATs, and there was a 13-to-1 ratio of males to females in there. And when it was last studied, a few years ago, it's down to a 3-to-1 ratio. Oh, that's obviously due to evolution over the last 20 years, because it's got to be due to genes, because it's... That's like saying genes explain the fact that in 1980s, people like wore pads on their shoulders and like power sneakers to work or whatever, and the fact that nobody does it anymore shows that the gene for wearing that on your woman executive decor deal has evolved, it's mutated. That's asinine. You do not go from 13-to-1 to 3-to-1 with a trait in 20 years in a highly interbreeding mixed population, in other words, humans, and have this as a genetic trait. Even function at the extreme has squat to do with the genetics of gender. It's I want to know what society the person got raised in. I could care less what their chromosome is. If I want to know how good they're likely to be at math compared to the other person, tell me where they grew up. That's the most important thing to know. Interesting additional thing. The second most reliable finding in all of measures of cognitive aptitude thing that has a gender difference is, on the average, girls being better at verbal performance tasks than boys, women than men, all that sort of thing. So what's going on in those 40 different countries there? What you wind up seeing is it depends on which country you're in. It depends on that gender equality index. And what you see is in the worst countries there by these measures, Tunisia, who else was it? Tunisia, Turkey, and South Korea. What you see is, women get better scores on verbal tests than men. And as you go to the more equal places, the gender difference increases. In other words, you got one of these. The more gender equal a society is, the less of a sex difference there is in math capabilities, and the bigger the advantage is for females over males in verbal performance. It's got everything to do with what society you're in. This gender difference in this realm of cognitive skills means next to nothing. And if I had any technological skills, I kept saying I was going to bring the figure from this paper. I'll have it posted, because this one figure, it's across all these different, you just look at it and there's the answer. It's got nothing to do with the genetics of gender difference. Okay, so what else would one want to emphasize here? Okay, what else would one want to emphasize here, which is this is a total mess and totally complicated. Okay, what have we gotten to so far in this field of behavioral genetics? There's all these different ancient ways of inferring something, twins, adoption. There's the much more modern ways, finding the actual gene. That's wonderful, that's exciting. Nonetheless, over and over and over, environment gets understudied. Environment is far more subtle than you think. Environment starts earlier in life than you think. And when you do it in a very formal, quantitative way, analyzing what genetic influences are about, you discover that scientists study things under circumstances where you constantly underestimate the importance of environment, blah, blah, et cetera, et cetera. But at the end of the day, don't genes have something to do with something or other going on with this? And obviously, they have some very important roles. You have genes that completely determine aspects of behavior, single genes that completely determine your intelligence if you're a housefly. Lots of interesting studies there. Single gene determination, a lot of these behaviors, and it doesn't matter sort of what the economic opportunities are for houseflies in that country versus humans. What you see there is, there are realms of behavioral biology where genes play very, very strong roles. And we will certainly see some of them in the lectures to come, but with two qualifiers. The first one we heard the other day, which is even when, with all of these criticisms on board, and you've got every critical tool to slice up anybody arguing that here's a high probability important genetic component, blah, blah, etc., you've completely cut it to pieces, and there still is a gene that seems to be doing something important for trait X. What we heard about the other day is nonetheless think about if there is an indirect genetic route for getting there. Is there a gene for extroversion, or is there a gene for your height and how people of your height are treated? Is there a gene for picking at grubs, or is there a gene for if you're really tall, you pick at grubs and people don't make fun of you? Is there all of that business about indirect genetic effects? So even when you see a gene for something, you have to begin to ask to make sure that you are not, in fact, looking at an indirect effect. Final qualifier, when you look at the really interesting genes in terms of what they do, when you look at some of the ones that have the most to do, say, with what differentiates our genome from the chimp genome, when you look at things like juggling your DNA just when you're making new neurons, what you see over and over is what human genes are about most dramatically is coding for ways in which you have freedom from the effects of genetics. And that is going to be a theme endlessly in the lectures to come. Okay.
For more, please visit us at stanford.edu. at Stanford University.
We push off into new terrain here, yet another bucket. But before doing
Ah, the failed protector of the Dark Dragon. Must I take your life yet again?
Where is the Dark Dragon?
It is in the hands of the Holy Vigor Emperor, your true master. You see, young Ryu, you are not so different from us. Those of the dragon lineage like yourself draw their blood from the same ancient deities as the fiends. You, most of all, should know that your true place is at the Emperor's side. My hatred will not be so easily defeated. I will bring about the awakening. Pitiful woman. Your sister must be quite precious to you.
Fiendish awakening.
He's wandering around here somewhere, My Excellency. Who knows what he's up to?
And the Dark Dragon?
To put it simply, it seems that the Dark Dragon, now free of its seal, is gradually absorbing the chaos of the battlefield and becoming a truly evil blade. It is not surprising that we did not know its true capacity, as the ancients were so careful to conceal it. Now, it is a weapon of extraordinary power, the likes of which have never been seen throughout human history. Whoever possesses it...
Do I sense greed in your voice?
Of course not, Excellency.
Good. That is wise. You will be rewarded handsomely, I assure you.
welcome.
You're still open?
Do you think I would close up shop over something like this? What can I help you with? I've been wondering, what is that sword you are using? Ah, let me see. Ah, this is a fine example of one of the swords of the dragon, carved out of a dragon's fang. It has the potential for unfathomable spirit power. But this sword alone will not do. There is a place to fit a jewel here, you see? A curved jewel carved out of a dragon's eye. With it in the right hands, this sword will prove more than a match for the Dark Dragon blade.
The Dark Dragon?
Of course I know of it. I have seen and heard of all manner of swords, both ancient and new, from east and from west. Now, if only we can find the dragon's eye.
Now the time of the awakening is truly at hand. This is not over. Welcome to the hellish depths of the Fiend Realm. The question is, can you defeat me? Receive the curse. Become a fiend.
You are Ryu Hayabusa. Over the corpses of my Black Spider clan brethren, I was able to make it this close to the core. But I cannot go on. Though it dishonors me, I give you this. Go to the core. You must reclaim Vigor. The equilibrium is collapsing. Master, I have failed. Forgive me.
What will he discover at the end of his journey? Appeasement for his vengeful heart? Or something else? The true battle is only just beginning.
my servants returned from the anthropology to take care of this puny insect once and for all. You're not as weak as I thought. That's it. It's time for me to take you on personally.
I expected nothing less. However, the power of the Dark Dragon is wasted on you.
Don't you see? The Dark Dragon is now truly an evil blade. His Excellency, the Dark Disciple, has been waiting for this very moment.
You've done well, Gamoth.
Huh?
Mirai.
The Dark Dragon is mine. No objections, I presume.
It's over.